And there we go. That's exactly the message you would receive. So if you do not wish to support that, unfortunately, the session will shut down for yourself if you do not accept. Now, I would like to first of all hand over to my co-host of today, and allow herself to introduce herself, and that would be Laura Viviani from AFSA.
Thank you very much, Kirsty. Hello, everyone. I'm Laura Viviani. I'm the Director of Biologicals within Humane Society International, and I'm following the activities of the Animal-Free Safety Assessment Collaboration. I'm super happy to see the incredible amount of representatives today. We have a strong representation from almost all over the world, with experts from industry, regulatory, and international organization. We are all here to discuss about the deletion of the abnormal toxicity test, and the fact that we are all here together is really an important sign that we all are interested in the change, and we are all here to collaborate together in order to understand how we can do that. It's, as you know, the abnormal toxicity test is an obsolete test, and it doesn't prove anything about the safety and the quality of biological products. So we are here again, thanks to you, to make sure that we can delete this test at the global level. So thank you so much, and I wish you all a very good workshop. I hand over again to Kirsty Reed, my colleague. Before that, I just wanted to thank you also very much, the International Alliance for Biological Standardization, for their support in the organizing of this meeting. So thank you, and Kirsty, it's for you now.
Thank you so much, Laura. And I just want to reiterate your thanks to those online today, and also to those who have helped with the organization of this meeting. For myself, it's an absolute pleasure to host this meeting with you today. And just to give an overview, and before I start, I just want to invite all the participants. You know, as has been said, we are representing a number of different countries around the world, and also different functions. And I invite you to just note in the chat for now which country you're from, or which village, town you're from, so people can see the variety of participation today. So, and it'll also give you that opportunity to try out the chat function. For myself, I represent EFPIA, the European Federation of Pharmaceutical Industries and Associations. And we believe that this workshop is very timely and is an important global issue. So we welcome the participants here. We're representing a global community. And within EFPIA, our members are, we are European based, but our members are global organizations. And this topic is one of great interest to them as global organizations. We take into account the discussions that will be taking place today, and we invite you, as I've mentioned earlier, to please put forward your questions, your comments. Also let us know during the meeting, also as follow-up to this meeting, of any information you would like to share with us that we can take up in our formal conclusions and possible report that will come out of this meeting. So on that note, I'd like to therefore take the opportunity. We'd like to try and show the video that was prepared of the ATT, sort of as a kickoff to this meeting today. And as I do so, at the same time, I'd also like to remind you to have a look in the chat amongst all the names of people and the countries they're representing, to also look for the different links to the video, which is available in a number of different languages.
So how do you find your ideal clients between people? Introducing Lucia.
We'll have to just get through the ads.
Abnormal toxicity test was developed in the 1900s to quantify phenol-derived preservatives as a quality control test for diphtheria serum. the ATT was also known as the General Safety Test or the Test for Innocuity. The test was first carried out on guinea pigs and later on mice. By the 1950s, the ATT became the standard test globally for detecting contaminants and toxins in finished biological products. It was adopted into a host of regulations worldwide, but as a general safety test rather than for the original specific purpose. In the 1980s, a significant shift began in the scientific consensus on this test. At the same time, the production process for vaccines and other biological products made great progress in preventing contamination and ensuring safety. 1996 was a turning point. Several key studies highlighted the inherent limitations of the ATT as not specific, lacking relevance, and not reproducible. These findings demonstrated that the ATT had become obsolete. This began a long regulatory journey towards the deletion of the ATT worldwide. 20 plus years of data analysis, expert reviews, and discussions. The ATT was deleted from the United States regulations in 2015. WHO recommended deletion for all products in 2018. Europe deleted the test in 2019 and Brazil the same year, with only one exception. It was deleted for almost all human vaccines in India in 2020. Japan has waived ATT for some products. It is no longer performed in Argentina, Canada, and South Africa. Around the world, many different stakeholders have been hard at work in the last decades to achieve global deletion of the ATT. Success is in sight, but there is still much work to be done. We need all stakeholders to come together again to plan a strategy for accelerating change. Through cooperation, collaboration, dissemination, and trust, together we can erase the abnormal toxicity test once and for all. Please join AFSA and EFPIA for a multi-stakeholder international discussion on October 14th, 2021.
And with that note, I'd like to hand back to Laura.
Thank you, Kirsty. We are actually going to start immediately into the action. So we are having two important keynote speeches in order to set the scene of the following discussion. And it's a pleasure for me introducing you our first keynote speaker, who is Thierry Gastineau from Sanofi Pasteur. I'm going to quickly introduce Thierry because he has an incredible experience within the pharmaceutical industry, and currently is the Global Quality Head of Innovation, Culture and Engagement within Sanofi Pasteur. And he has 34 years of experience in the pharmaceutical industry, the last 19 within Sanofi Pasteur. He actually was the first IFMA representative at EPAC, so the Immunization Practice Advisory Committee at WHO, within 2010 and 2012. And starting from the 2019 in his current role, he actually is member of an important list of groups and organizations. Within Vaccine Europe, he is the co-leader of the CMC Quality Core team and the ICH 12 task force as a leader in that group. Within IFMA is the leader of vaccines head of quality group, and within again Vaccine Europe and IFMA is the leader of the CMC GMDP COVID-19 task force. Is member of the US PDA, so the Parental Drug Association, and also member of the One Voice of Quality group. He is a pharmacist and he holds a postgraduate degree in molecular biology, both of them in France. Thierry, I leave the floor to you, and welcome, and thank you very much.
Thank you, thank you, Laura. And first of all, I'm very honored. To have received your invitation as a keynote speaker to this a great conference. So thanks a lot for the invitation. So actually, my talk will be about speaking about the global challenge of post-approval changes. So that's what I'm going to put you in the picture of. And of course, I will make a connection with the ATT test, but I'll start actually to discuss more widely about the challenge that we're meeting with post-approval changes. Next slide, please. All right, so as I guess you all know, after initial licensure of a product, of a drug, pharmaceutical product, including vaccines, of course, so a lot of post-approval changes are needed to maintain actually the life cycle management of the product. And there are multiple reasons for that. Actually, and you see here in the picture, I mean, something which a picture that was actually presented or published, sorry, in a publication. And there are multiple reasons which are related to routine changes, continuous improvement, innovation changes, or just changes that are needed to ensure the appropriate supply chain is sustained over time. Now, for vaccines, actually, those post-approval changes are pretty complex to manage because of several factors. The first one is that, as you know, the vaccine manufacturing is a complex process. We're using a lot of equipment, a lot of raw materials, and there is a huge number of testing activities. Roughly, for a standard vaccine, 70% of the activities are related to testing. Also, we are dealing with some complex vaccines, in particular those for which we have combination of multiple antigens. I give here just one example for the polio antigen, and actually one post-approval change may impact more than 500 licenses across the world. And last but not least, also facilities and processes are aging, and also we need to continue improving our product and process knowledge, also with regards to new technologies and so on. So a lot of complexity around all this, if you look at this from a product manufacturing and control perspective. Next slide, please. Okay, now if we're looking at the regulatory landscape, the first thing that you can see actually on the left-hand side is that from a company's perspective, well, our companies, for most of them, are globalized, which means that our target and what we're doing is that we're manufacturing and controlling one product for one world. And when I'm saying one product, it means also one science and hopefully one dossier for one world. Now, if we're looking at the regulatory landscape, how it works, actually regulatory approvals are nationalized. What it means is that every single authority in the world, roughly, is managing the regulatory licensing process on a national basis. Of course, there are some specificities, for example, in the EU, where we have some harmonized processes. But this is basically what this regulatory landscape look like. Next slide. Now, the point is that everything could be good if we were not in the situation where actually not all countries in the world have the same legislative framework, the same regulatory requirements, the same regulatory procedures, the same and predictable timelines, the same scientific and regulatory maturity. So imagine, and you'll see that also in the following slides, is that we are dealing with a still highly heterogeneous regulatory worldwide landscape. Next slide. So what does it mean if we deep dive a little bit on the post-approval change perspective? Now, if we take here again, depending on where you sit, if you are sitting at the level of a regulatory agency, what you see from a given manufacturer is, for example, one post-approval change for which you receive a dossier, you assess the dossier, and hopefully you approve this dossier, this variation dossier. Now, if, like me, for example, I'm sitting in a pharma company, this single post-approval change, I have to submit it in numerous countries across the world because of the reasons that I've just indicated before, because actually there is no single harmonized system, regulatory system all across the world. So the same dossier for the same post-approval change is submitted in numerous number of countries across the world according to their national regulations. Next slide. The point is, if you remember well, the very first slide that I presented, there are multiple reasons why actually we need to manage multiple post-approval changes across a single year. And actually what I described just before about one single pack that we need to manage in multiple countries, in fact, this is you need to multiply that by as many post-approval changes that the companies have to manage on a yearly basis. And I have also to indicate that this picture that you see here is even more complex. I'm sorry, but I didn't find a way to make it even more complex. But in particular for those post-approval changes, which are impacting multiple combination vaccines. Remember the example that I gave you at the very beginning with regards to the polio antigen, for example, which is impacting multiple licenses. It's because actually it is entering into the composition of multiple combination vaccines. So a dramatic amplification for all these reasons. Next slide. So this is a real illustration. This is not from my company. You see the source. This is coming from GSK Vaccines. And actually, what I'm describing to you, by the way, is not something which is specific to Sanofi Pasteur, but this is something which is actually shared across all vaccine manufacturers. So this picture describes something that I need to explain a little bit so that you understand what it is about. Each column corresponds to a given country. You don't have the names of the countries here. It's not possible. Each column equals a country. Each line equals one single post-approval change. So it means that, and you see here the color coding at the bottom. For example, in green, it means that no variation is required in the country. In yellow, it's notifiable change. In red, prior approval, and so on. So what it means is that when, and this is, I think, the view of a yearly analysis of what was managed by this company for all the post-approval changes, they had to manage for their vaccine portfolio. And you see here, I mean, this beautiful but very complex picture of colors, which illustrate how heterogeneous is the situation, with one exception that you can see in the middle, which is Europe, where you see that actually it's basically roughly the same for all countries in Europe, because indeed this is maybe one of the region in the world where there is the most harmonized regulatory processes for managing licenses and post-approval changes. So the ideal scenario would be to have something which is a bit similar to Europe, but not, I mean, this constellation or this patchwork, if you will, of different colors, even if it's beautiful on the paper here on the slide. Next slide. So what is the outcome of this tremendously complex situation? Before I speak, by the way, about the outcome, what also you need to understand is that because of this one aspect of this, which is specific to this heterogeneity, is also the timing. I said that actually not all regulators in the world or regulations in the world have the same timings, and even not predictable, I have to say. And actually, for one single pack, and multiply that as many packs as I indicated before, it can take up to 4 years and more to get approvals of those post-approval changes in all countries in the world. I'm not saying that it takes 4 years in all countries. For some countries, it's very, very fast. But the latest countries in the world who actually review and approve can take up to 4 years. And you have the whole range of timing in between. The second challenge here is that from a manufacturing point of view, we cannot manage two versions of the same manufacturing process in the same facilities and we cannot manage control processes in parallel. I mean, we don't have, imagine that if we are making so many changes, if we want actually to accommodate all types of situations that you have seen in this beautiful picture before, that would mean that we have to multiply our manufacturing and control assets by as many variants or variations of your product pre and post change, depending on the different situations. So what we are doing to accommodate this situation from a manufacturing point of view is the fact that we are constantly juggling with this very complex situation by managing and trying to accommodate multiple elements, multiple criteria. We are taking into consideration the dossier submission dates that we submit in the different countries in the world, which may be different. It's not for all countries. Sorry, we cannot submit to all countries at the same time because also of different regulations. Also, we take into consideration the different approval timings, as I said. Also, the different implementation dates. Also, we take into consideration the supply demands and forecasts, which are, by the way, pretty much constantly evolving. And of course, we are building hopefully appropriate inventories to manage this tremendously complex situation, while of course remaining compliant with the licenses. So very, very complex situation that we try to manage the best we can in our companies. But the outcome of this is not a timely and equitable supply to all populations of vaccines of the same high quality. All vaccines, of course, are good quality, but they are not actually supplied with all the changes at the same time to all populations, and running a risk of shortages of vaccines due to this type of situation. Next slide. Right, now that I have explained to you, this complex situation, what we need to acknowledge is that some good progress have been made, but there is still a lot to do. And I've just cited here a couple of them here. The first ones are related to WHO, and I would like really to acknowledge and to thank the tremendous efforts that actually WHO is doing in that respect. I've just cited here just a couple of elements. There are many others, but some which are pretty important in the context are actually guidelines on post-approval changes for vaccines. One which is much more recent is the good reliance practices, which have been published back in March this year by WHO, so that actually we avoid redundant activities across different regulators, and also the network of national control laboratories. So a lot of very important elements, and hopefully that all countries in the world can be inspired or apply in their national regulations. Of course, there are also all the tremendous work which is done at ICH level. I do know that not all countries in the world, and maybe some who are in attendance today are not ICH members, but still this is also very important in terms of harmonization. There are also some bodies, regulatory bodies, if I could say so, which are very important. I'm citing here a couple of them which are embracing multiple countries in the world, such as ICMA, ICTRA, the IPRP. You have all the significance here on the right-hand side. So those regulatory bodies are also doing a fantastic job in order to foster harmonization, reliance principles, and so on. And of course, last but not least, there are some regional reliance and recognition mechanisms which do exist. It's not only in Europe, there are also regions in the world which are actually applying those mechanisms. Next slide. Now, actually, as you said, Laura, we have a task force at EFPIA working on COVID from a CMC perspective. And from our first analysis of the COVID-19 lessons learned for managing post-approval changes, we clearly identified 4 essential elements here that we do believe and we would like actually to see implemented, not only for COVID, but also in the routine management of post-approval changes. I'm not going into the deep details, but there are all, because I'm pretty sure that we're going to discuss that into details in the rest of the conference. But there are about science, adopting science and risk-based approaches. This was a critical element actually to put vaccines on the market in record times against COVID, but also for managing post-approval changes. Convergence and harmonization is absolutely key, actually, to get rid as much as possible about this complex picture that I've illustrated before. Regulatory reliance was something which was I mean, a key element which was discussed a couple of weeks ago at ICDRA level. And actually, we need to actually move forward with more reliance across countries. And last but not least, maybe a little bit more longer term is about digital. How could actually digital can help actually in managing post-approval changes? But this is maybe another topic. Next slide. Yeah, so here I have 2 slides about the abnormal toxicity test. So as we have seen, by the way, in this great video that you displayed at the very beginning, this is a story which actually started a while ago, 25 years ago, if we think about it. And actually, as indicated in the movie, it started back in, well, it started before, but at least there was a key milestone in 1996 when actually it was considered, and there was a scientific consensus indicating that this test is not specific, not relevant, not reproducible. There is a milestone also that I wanted to highlight here in 2010, which is very important, which Catherine Milne will come back in the second presentation on, is about the EU directive on 3R. And after that, we need to wait for 2015 until when actually, to start with, US FDA has actually removed this request or this requirement of running the ATT test. And as you can see, and I'm pretty sure you're aware of, a number of health authorities, actually, or regulatory bodies, have actually removed this test from their regulatory requirements, and a number of them are cited here. However, and I think that's the topic of this conference today, still, we're in 2021, a lot of other countries did not remove this test yet completely. Next slide. So what is, by the way, our Sanofi Pasteur experience in that respect with regards to the ATT test? So what is the challenge? There are multiple challenges, in fact. The first one is that this test is still in the QC profile of multiple vaccines, of multiple of our vaccines, as post-approval changes are actually still in progress, and this test is still required in some countries. Second is that even when the test is removed from some pharmacopoeias, some national regulatory agencies still request that the variation is submitted for a prior approval on a per-country basis, instead of submitting a notification, actually providing the pharmacopoeia is updated. Why not just submitting a notification that would be an accelerating factor for actually removing the test? And when exemptions are requested from our companies, some regulatory authorities accept and some others don't, because they're all asking a lot of other justifications and data required, whereas actually the consensus was already reached. So it creates a huge regulatory international complexity due to the absence of convergence and harmonization despite scientific consensus that was reached years ago, as I indicated. Absence of reactivity from the national regulatory agencies for removing the test from their pharmacopoeias. You see that, as I indicated, US FDA started about six years ago. Absence of risk-based approaches, as variations are still requested while the test is removed from some pharmacopoeias. And absence of reliance based on approvals granted by stringent regulatory authorities. So what is the impact of this situation is that we have a continued burden of meaningless. So you said, Laura, in your introduction words that it is an obsolete test, sorry. So yes, I indicate here it's meaningless actually to continue running this test for both companies and local authorities, according to us. It is a continued use of animals, which is against the 3R directive that we know in Europe. And it also has a delayed, it has an impact also, it delays the supply if the test is continued to be performed by the manufacturer and or the national control laboratory upon importation. Next slide. So to finish with, it's a little bit small on my screen, so I hope I will succeed reading it. But so I invented here a new concept, which is the 4R concept, in addition to the 3R. The 3R being reduce, refine, replace. So here I'm adding here rely. So actually, there are a number of things that actually we do need to consider with regards to effective management of post-approval changes. By refining, I'm not going into the deep details. Anyway, I cannot read the sentences, but which are about actually refining the processes. That's one thing. Reducing also the number of post-approval changes that need to be submitted, and also reducing in vivo testing. Replacing national procedures and also in vivo testing. And like I said, the new one that I added to make it a 4R is to rely. And like I said, this was a keyword that was cited during ITRA, and rightfully. So that actually authorities in the world do make reference and rely upon what has been already accepted or reviewed and accepted by other authorities in the world. So with that said, I think that's my last slide. So that's the conclusion here. I just took a couple of verbatims that you can see from the Dr. Socorro Gross-Galiano from Brazil that I leave you reading. And I'm reading here what was said in the last recommendations of IGDRA, which took place 3 weeks ago, where it says that actually NRAs should apply more harmonized systems for all post-approval changes throughout a medicine product life cycle and reliance models with a risk-based approach for PACS evaluation. Next slide. Yes, thank you. Thank you very much for your attention. And you can see here that despite the fact that it was a Sanofi Pasteur presentation, this is a collective effort also from other companies which do participate in a number of working groups working on the topic of post-approval changes. So thanks a lot for your attention.
Terry, and thank you to you for an excellent presentation. I think it really kicks off, it's an excellent keynote to kick off discussions that will take place later. So I'd like to move on quite swiftly, but first, before I say that, I have an extra R, and that is remove, and that's, I think, what we're looking at today, is to have the removal of the ATT test from all global pharmacopoeia. I'd like to introduce Catherine, and now Catherine Milne is representing us from the Edqm. So she received her doctoral degree from the University of Toronto Department of Molecular and Medical Genetics, and was a postdoctoral fellow at the Medical Research Council in the Laboratory of Molecular Biology in Cambridge, in England, in the field of molecular genetics and developmental biology. She then joined the Council of Europe's European Directorate for the Quality of Medicines and Healthcare, which we all know as Edqm, in 1999. And she is now head of the section of Biological Standardization, International Standards for Antibiotics, and the Official Control Authority batch release of human vaccines, medicinal products derived from human blood and plasma, and veterinary vaccines, respectively. Just quickly, in her role as the Edqm Biological Standardization Program Administrator, she coordinated a number of projects for the standardization of methods and reference materials for the evaluation of human and veterinary vaccines and medicinal products derived from human blood and plasma, with the focus on the 3 Rs. And she represents Edqm as an observer in the EMA's Biological Working Party and the Committee for Advanced Therapies at the EMA, and also in their 3 Rs Working Party, and also at the International Alliance for Biological Standards, VBC. So with that, I'd like to hand over to Catherine, welcome you, and thank you for joining us today.
Well, thank you very much, Kirsty, for this kind introduction, and thanks also to the organizers. It's both an honor and a pleasure to be here to talk about this important subject today. So if we can go on to the next slide, please. This is just the title of my talk, and I'll be guiding you through the removal of the ATT from the European Pharmacopoeia. Next slide, please. Here we have just a quick overview of the elements that I'm going to present, but I won't go into detail, as you'll see as we go. So we can maybe go on to the next slide. So here is just a very brief history of the Edqm and the Council of Europe's commitments to 3 Rs. For those of you who don't know, the Council of Europe is situated here in Strasbourg. It was established in 1949. It's one of the oldest pan-European organizations. And to date, there are 47 member state countries and 6 observer states. And the focus of this organization is human rights, democracy, and rule of law. The Council of Europe gives the potential to create different conventions that could be signed by the different members. And in 1964, the European Pharmacopoeia Convention was developed as an international treaty. To date, there are 39 member states, including the EU, and 30 observers, including the WHO. And the important thing to note here is that the European Pharmacopoeia texts are mandatory in all member states. So that means that the quality requirements established there must be implemented in all the member states. This leads to a harmonization of technical requirements for the authorization and manufacture of medicinal products. Now, if we jump forward quite a bit to 1986, the Council of Europe also developed a European Convention for the protection of vertebrate animals used for experimental and other scientific purpose. And this has really been the basis of a lot of the work going forward in the EDQM and the Council of Europe and elsewhere in Europe. And in fact, the convention was, many of the aspects of the convention were incorporated into the European Directive on the protection for animals that was developed in around the same period. 1991, we had the starting of the biological standardization program here at EDQM. This program is an agreement between the Council of Europe and the EU Commission in Brussels, and it's co-funded by these two organizations. We have several roles. One is to develop common reference material, reference standards for use with the pharmacopoeial methods. And one of our main focuses is the development of methods with a focus on 3 Rs, to reduce, replace, and refine the use of animals in quality control testing of medicines, and particularly biological medicines. 1994, a few years later, we had creation of the OMCL network. Presently, the OMCL network has 71 OMCLs in 43 countries. This is a network of independent laboratories within the EU and beyond with our some of our other members. And they're responsible for the independent control, physical control of all types of medicines, but also biologicals. And they have been a driving factor in going forward with the 3R programs that we have at EDQM and throughout Europe. And finally, Thierry has already mentioned it, in 2010, the Directive 2010-63 EU came into force. This is for the protection of vertebrate animals for scientific purposes. And this really set a framework, an overarching framework in EU legislation to put a focus on the reduction and removal of the use of animals. Next slide, please. So with respect to the pharmacopoeia and 3 Rs in European pharmacopoeial testing, this is integrated at many levels. We have the overarching texts of the general notices, which foster and enable the application of 3 Rs. This is done by encouraging the reduction of animal usage and allowing, in particular, alternative methods. There are the conditions for this are explained, and highlighting the consistency approach, which also has an impact on the ability to apply 3 Rs. We have the general monograph for vaccines for human use, which highlights again the European Convention and the idea that the minimum number of animals and causing the least pain and suffering and stress has to be taken into consideration when implementing and developing any test. And then, of course, we have individual monographs that encourage the use of 3R alternatives in different ways. The use of humane endpoints, for example, when it's unavoidable to use animals. We have detailed protocols for some validated 3R methods that are included in the specific monographs where available. And then we also have what we call door openers, where if there's not a specific in vitro test described, it makes it clear that this option is a possibility if appropriately validated tests can be presented to the authorities. Another important chapter in the pharmacopoeia is 5214, and this is a fairly recent chapter as covers the concept of substitution of animal tests. Now, we all know that the gold standard tests are gold standards for some reasons, but not gold in other ways. And one of the problems is always the variability of these tests. So it's often very difficult to compare directly alternative and in vitro alternative to the original test without revalidating, in fact, the original test. And it's also sometimes the case that one in vitro test may not be sufficient to replace an in vivo test. You may need a combination of testing approaches to do this. So this chapter helps to outline this concept. And I think it's very important for going forward with this work. And finally, the European Pharmacopoeia has an ongoing review of all of the European Pharmacopoeia texts in line with scientific understanding, and of course in the context of 2763 to employ 3 Rs, and to take the 4th R, which we heard about from Kirsty, the removal R. Next, please. So the abnormal toxicity test, we've already seen a nice video about it. I don't think we need to go into a lot of detail. You know, it's known by several different names: the general safety test, the innocuity test, and the formats vary in the different pharmacopoeias. But the main principle is that material from the batches of a product are injected into guinea pigs and or mice, and the batch passes the test if animals don't show signs of illness or die within a defined time frame. So this clearly leads to quite a bit of animal suffering. There's a considerable usage of animals for vaccines. As an example, there are 5 mice and 2 guinea pigs. Somehow the 5 disappeared. 5 mice and 2 guinea pigs for each batch. And increasingly, there has been a questioning of the scientific usefulness of these tests in general. So this was one of the most controversial animal tests in the European Pharmacopoeia, and it was a priority target for reevaluation. Next, please. Now, here's a little bit of background of the ATT, which we've also already seen in the video. So I'll just go through quickly. This test has been around a long time. Dates back to the early 1900s, even the 1890s for the mouse test. At the time, definitely state of the art. It was useful and saved a lot of people from products that were of insufficient quality. The mice being used to detect levels of phenol in the early sera products, initially diphtheria and tetanus, and later for the inactivated bacterial vaccines. So the mouse effectively being used as the readout for the phenol content. And then there were, for the guinea pig test, this was linked to trying to deal with incidents of contamination of antisera with toxin spores in the early 1900s in the US. So the guinea pig test was introduced as a biological indicator for extraneous clostridial toxins. And this was used for sera initially, but then later vaccines. And then these 2 tests had their sort of independent life early on, but in the 1940s, both were combined to become a general safety test. And the ATT, so the test with the guinea pigs and the mice, was mentioned as such for the first time when the WHO began developing international guidelines after the Second World War. And it was introduced eventually into almost all general testing requirements for immunological and biological medicines, both human and vet, around the world. So the ATT has remained largely unchanged since then, despite the evolution of analytical techniques and manufacturing processes up to 2021. Next, please. So the work of the Pharmacopoeia, moving towards removal. As you've seen in the 80s and 90s, there was an increasing sensitivity to 3 Rs, and the ATT was a clear target. So how do we deal with the ATT, which was used historically as, if I can say, a catch-all to make sure that there was nothing bad in the vaccines or in the products, but the purpose and validation parameters were not clearly defined. So this led us to a conundrum, because to refine, reduce, or replace, we talk about alternatives and having to validate against the existing test, but this really was a casse-tête, not really possible. So another possibility would be to remove the test, but this was a very big step, or considered a very big step at the time. And regulators and QC experts at the time were acknowledging the issues with the science, but stressed that it had been used successfully in the past, and it was relatively low severity compared to some other tests, and the number of animals wasn't so high, and they were reluctant to take the leap. So to help move things forward, the Paul Ehrlich Institute, supported by the German Ministry for Education and Research, did a very extensive review of data and a retrospective analysis in between 1994 and 1995. And this analysis concluded that the ATT was neither specific, reproducible, reliable, nor suitable for the intended purpose. And as an additional point, there were more relevant tests available for testing phenols and toxins currently available. This, combined with the fact that the use of GMP and stringent QC measures to prevent contamination, put into further question the relevance of the ATT. So next, please. So going towards removal after reviewing all of this extensive review, the European Pharmacopoeia looked to deleting from routine batch release testing from greater than 80 monographs, the ATT, and this was done in 1988. So as of 1988, there were no more routine tests for ATT in the Pharmacopoeia. There was a complete deletion accepted for veterinary products. There was different approaches for the different human products. There was complete deletion for sera and immunoglobulins, complete deletion for DTP, and for all other products, there was deletion at the routine batch release test, and the test was moved to the production section, so it was used during development. This in itself led to a significant reduction in the unnecessary unscientific use of animals, which improved the situation for the ATT, but it still left the ATT in the Pharmacopoeia. And since that time, hundreds of thousands of lots of sera, immunoglobulins, vaccines for human and veterinary use have been used and released in the EU without any noticeable problems. Next, please. So time moving forward from this stage, there was still a focus on ATT. And in 2015, the European Partnership for Alternative Approaches to Animal Testing held a very important workshop that took an in-depth look at the ATT issue with global stakeholders. The workshop included review of case studies and data and brought together many experts in the field. And the conclusion of that workshop was that the ATT, again, same conclusion, lacks the scientific relevance and that its emission does not compromise the safety of biologicals. So they came to the consensus that there should be action for deletion of the ATT from regulatory requirements worldwide, and that this should be done at a global level and with preference using a harmonized approach by all regulators across the globe. And this would be important to make the deletion really effective. And I think Thierry's presentation has shown us some of the complications that can arise when there is a disharmonization. Next, please. So after the workshop in 2015, the European Pharmacopoeia went to work again and took this as a basis for the review of the remaining 49 monographs where the ATT was still included. There was a detailed evaluation conducted for each monograph with notes on the different monographs in the relevant groups. It concerned a range of products, human vaccines, antibiotics, antifungals, allergen products, even sterile plastics and containers for human blood. And of course, the general chapter 2.6.9, which was the chapter that described the ATT. There was a decision to suppress the ATT at the November 2017 Commission session. The revised monographs omitting the ATT were published in July 2018, which meant that it came into force in January 2019. And this revision of the monographs included a simultaneous suppression of the chapter 2.6.9 Abnormal Toxicity from the European Pharmacopoeia. So next point, there's absolutely no more ATT left. Next slide, please. In the European Pharmacopoeia, which was a major step forward for 3Rs, and I might add a major step forward for the scientific rationale. Next, please. So the European Pharmacopoeia and beyond, we've heard the importance of the global impact from actually earlier than 2015, but 2015 more formally. Edqm presented regularly at the WHO Expert Committee for Biological Standardization plenary sessions a request to consider the removal of the ATT from WHO guidelines with presentations and updates from my former colleague Karl-Heinz Buchheit, who I think many of you know. In November 2018, ECBS, WHO, and the stakeholders made a proposal to discontinue the test in all guidelines, and this led to an ECBS recommendation to discontinue it in guidelines on vaccines and biologicals at this ECBS session, which was another further step forward to global acceptance. Unfortunately, the texts remain in the guidelines because of the way the guideline updates work at WHO and are to be reviewed when next, updated when next reviewed. And I know that there's a big undertaking underway to have a look with respect to 3Rs in all of the WHO texts. So hopefully this is something that can be taken up in that. I think that's being led by NC3Rs. This fact that they're still in the guidelines is overridden by an ECBS statement but of course, the next step is to update the text. And if you click the next slide, please, you can see this is the outcomes of the ECBS meeting, and highlighted in red is the statement that the innocuity test will not be included in any future guidelines, but there's a clear indication made in the report that the inclusion of the tests in previously published WHO technical report or series documents should be disregarded. Next, please. So before leaving, I wanted to just mention a few other activities in a similar vein in the European Pharmacopoeia. You see here the target animal batch safety test, which has a similar parcours to the ATT. Here we have an innocuity test of inactivated or live veterinary vaccines by administering an overdose of every batch into the target animal. And here we went through a process also for similar reasons, beginning in this case with waiving in 2004, the test upon showing data, and then finally in 2013, removal from 80 monographs and general monograph and 2 chapters. And we see that VICH guidelines 1555 are also moving in the same direction with harmonized criteria to waive the TABST for these 2 categories of vaccines. And this is available since 2018. Next, please. A few other points of recent progress. And as I mentioned, we're continuing to review especially the older texts to make sure that they're up to date in a scientific context. In all cases, there's a careful review by the groups with respect to rationale and the historic evidence from vaccine lots. The first example is a histamine sensitization test used in acellular pertussis vaccines. Here, this was stimulated a little bit by a biological standardization program to standardize the CHO cell-based assay for residual toxin. And in the development of the program and the method development, the text was reevaluated. And in fact, the revisions went further than anticipated. So the residual toxin by standardized CHO is now done only on the non-absorbed purified pertussis components that have been detoxified using a validated procedure. The irreversibility of toxoid test has been deleted, and there's no more toxicity testing on the final lot because, of course, the more sensitive test has been used upstream. And this is effective as of the 1st of January 2020.
Catherine, just to say we're running very late.
Okay, well, the next 2 examples also show how this similar process has been done for tetanus and diphtheria vaccines. And the last slide, I think, is just to say that the Edqm and the European Pharmacopoeia Commission, its experts and stakeholders are all committed to the principles of good science and to supporting the directive, and that we continue to do work. And I wanted to end with a special mention to Klaus Kusler, formerly of the Paul Ehrlich Institute, and his colleagues for their dedication and effort to use science and evidence to support the relevant change for ATT, and Lucas Bruckner for his similar efforts in TABST. Thank you.
Thank you, Catherine. It was very useful introduction for our audience. And in the interest of time, I would like to introduce our next session. But I would like also to invite you to put any questions to both Catherine and Thierry into the chat. So I would like right now to introduce the moderator of our next roundtable that is called Global Perspective on the ITT Deletion, Sajan Shahid, as a PhD in biology with a focus on immunology and host pathogen interaction. Before he joined GSK Vaccines, he worked on the diagnostic of zoonosis disease at the German Federal Health Robert Koch Institute. In his current role in global quality in GSK Vaccines, he is leading the company's strategy to reduce animals by substituting them with state-of-the-art technology. Sajan, welcome. And
thank you.
I would like also to welcome all the participants to this roundtable to unmute themselves and open the video. Thank you.
Thank you, Laura, for the nice introduction. And yes, good morning, good afternoon to all the audience. I have the pleasure to moderate the first roundtable. As you will see, the focus is on where the ATT has already been deleted in certain regions, and we have a few questions prepared. However, we don't want to limit the discussion on the prepared questions, so don't hesitate reach out via the chat if you want to speak up and ask a question, or if you prefer rather to write down your question, please introduce quickly who you are. And if you like to ask one of the panelists directly, then please indicate also the panelist. Otherwise, we give a very short introduction about our 6 panelists and not go too much into detail because the time is really short. But as you can see here, we have Mark from Janssen Vaccine, we have Lina from the Immunologicals Limited from India, we have Dian Ling from the WHO, Dean from Health Canada, Robin from the FDA CBER, and also Philip from the Bill and Melinda Gates Foundation. So welcome to the first round table. Please turn on your cameras. And already have a first opening question before we hopefully get more out of the chat is, if you think about the abnormal toxicity test and the parameters that need to be in place to ensure patient safety, is there something that you see beyond the endotoxin test and the sterility test for biological products? This were one of the questions asked up front from the audience. If someone likes to take it.
Good afternoon and good morning to each and everyone. So my name is Lina, and first of all, I would like to thank each and everyone. And for this question, I would like to answer. So the additional test, whatever endotoxin sterility test, are important to evaluate the safety of biological products. And we have various physicochemical tests as mentioned in WHO pharmacopoeia to assure the product safety. However, the abnormal toxicity in terms of biological response as that is getting provided in the ATT test may not be provided. However, need some enlightenment on this thought.
Thank you, Lina. And maybe I can ask Dean or Robin or Dian Ling from a more health authority perspective to give their view.
Yeah, Shajian, I'll chip in as well. So there is a vast array of tests to deal with the safety and control of products has been described by our earlier speakers. And the notion that we could rely on the ATT as the safety measure for vaccines or other products is simply not relevant, as has been pointed out. You know, complex antigen products go through hundreds of tests before they reach the end of their production cycle and go through lot release, both through the manufacturer and through regulatory authorities. And they are, each of those tests is designed for a specific purpose, but many tests are such as the endotoxin, pyrogenicity tests, if that's relevant to a product, but many others are involved in the testing. So it's an extremely rigorous process. And I will point out, rather than stating what is done, I just, again, cite an example of where the ATT, or the general safety test as it's also known, has sort of failed in its general properties. Is that, you know, some years ago we had the deliberate adulteration of heparin products coming out of certain regions of the world. And again, that general safety test did nothing to identify that deliberate adulteration of that product. Those products were admitted to patients and some died. Of course, that was a less than sensitive in vitro test that was responsible for controlling the potency of that product, and it was tricked by a deliberate effort to do so. The general safety test had no bearing on that whatsoever, and would have failed in many other situations where more sophisticated analysis, anything from HPLC analysis to many other robust technologies, would have detected some impurity in the product and flagged that lot for investigation at the very least. So the notion that we rely on a simple, on the one hand, complex and black box test like the ATT is just not borne up by any science, and has been described by Catherine and Thierry. It has no scientific basis as a methodology in the control of products. Over.
No, thank you a lot, Dina. That was also a very interesting response, because as in fact there are some former publications stating that the ATT can detect impurities. And so I think the argument you just have explained is basically showcasing the opposite. And indeed, unspecific assay might not be the best option if you need to look for something specific for a specific risk. Before I move to the next question, any other response? Someone else who wants to react to the question?
Maybe I can chip in a little bit.
Yes,
This is Daniel Lam from WHO World Health Organization, where we're developing the international standard guidelines for biologicals and vaccines. As you know, WHO has been providing international standard and the guidelines to the member state to assist the country to develop their national document or legislations to ensure the quality, safety, and efficacy of the product. Today we're talking about the ATT. WHO has been working on the deletion of this test from WHO recommendations from quite early. Before 1999, WHO, it's a recommended or required test on the lot release for biological and vaccine product. After 2000, WHO has changed its position, and to perform this test can be omitted once the GMP has been implemented and the production consistency has been demonstrated. Of course, with agreement with national regulatory authority. In 2018, WHO reviewed all the scientific publications and the debate on different occasions, and the ECPS, the committee, decided to remove this test from, or not continue to recommend this test for lot release of this of vaccines. Also, as you have been heard from Catherine's presentation, WHO is not to revise its recommendation regularly, but some of the tests have been published, printed out, but the published one should be disregarded in the printed out document. And we realized at the moment there are still some countries that are still requesting this test for lot release, for marketing authorization for biological products. And I think there is a long way to go, and a lot of work to do for WHO to implement the recommendations of the committee into national regulatory authority, and discuss with the regulators and to make awareness of the WHO position on this test, not only to regulators but manufacturers and the public as well, and really to at the end to get the harmonization or convergence of the regulations in different parts of the world. Thank you.
Thank you, Jianling. And I think it's really an interesting view, and maybe to follow up a bit on that, when we think about the COVID-19 vaccines, where we have several products from several companies, from several countries and regions of the world, do you see an impact there on the ATT? Do you see that overall shift to release those new vaccines without the ATT, or is it rather that it's based on the national guidance as it is in place for other life cycle vaccines, or whether the bold move to be fast and courageous and skip this test?
Yeah, of course, if we remove this test, it will save a lot of time and to access to the vaccines. The ATT test, it normally takes about 7 days. Have to get the weight, whether there is a decrease of the weight of animals, either it's both mouse and the guinea pigs. And to remove that, I think it's a WHO position, and because as we heard, this test had been introduced in 1900 at that time, and the confidence on the vaccine or the biological product mostly based on the test on the final product. Since there's no concept of good manufacturing process or validation during that time. Since the GMP has been implemented and the validation, comprehensive validation for manufacturing processes and sophisticated control of starting materials, and there is less and less introduction of any toxic component into the manufacturing processes. To remove the test, there is no compromise of the quality, the safety of the product. Now, talking about the COVID vaccines, because there is an urgent need globally, and even there are a lot of parts of the world that they have not access to the vaccine yet. So reliance, it's a WHO position. We recommend it globally. The regulatory authorities in the receiving country, if they don't have the capacity or the competency to review the product, and a good reliance or the reliance to the decision for regulatory authorities of the producing country or the competent regulatory authorities, it's what we recommended. If you rely on that, not only for the marketing authorization, but also for lot release of the product, also it's very important. I think the reliance on the decision, recognize the lot release certificate from the NRA for producing countries, it's what we recommended to the country where they receive the vaccines. Thank you.
Thank you, Yanling. And maybe, Robin, I can also ask you a similar question, because most of the vaccines, or a big part of the COVID-19 vaccines, have been developed or are produced in the US. And what is the perspective in the US regarding those novel vaccines against a novel disease and a very historic and old test?
So I was really surprised, actually, when I started preparing for this, to see that any COVID vaccines anywhere were being required to be developed using the ATTs. Certainly since we made the very unilateral decision and actually changed the Code of Federal Regulations to remove the test, I don't think at this point we have any products that that we have an expectation that the ATT is done even throughout early development. So we have moved forward and are reviewing, I think, almost 100 different COVID vaccines of a whole variety of different platforms, and we have not required the use of the ATT. Now, that said, we have, for certain new platforms, actually gone back and asked for some toxicity, some safety in the preclinical space as part of product development to establish and characterize the safety of the product along with the manufacturing process that will then assure, product that meets all of the regulations. So it's very easy for me to answer this question in the sense that we have not required the ATT for any of the many COVID vaccines that we've had under review.
I mean, that's really great to hear. And then the other, because working from the industry side where I myself are, was this also clear for the manufacturers, or were they still from the beginning rather in favor of including the data, ATT data?
So when we have manufacturers that submit to us a development program or submit in the pre-IND or space requests about tests that's required, if they have included that they will do the ATT, or in our case, we call it the general safety test, we actually tell them. We presume when we see that it's because they are talking to NRAs which still require that test. And in response to them telling us that they will be doing this test, we say this test is not required for any US development or licensure. So
it's quite clear. So a more
explicit
harmonized
about that.
view on that would have prevented the ATT test. Thank you, Robin.
Shahjahan, this is Gautam Sanyal. May I make a comment and ask a follow-up question on this? So do you know from the manufacturers and of countries that normally have required or have not removed the ATT requirement, NRAs, have made exceptions? I mean, I'm talking about receiving countries for vaccines that have been produced with new technologies or even old technologies coming into their countries. For COVID vaccines, have they made exceptions? Do you know? I mean, can that be a precedent? Although I know this was a pandemic vaccine, but at least for pandemic vaccines, could that be setting a precedent for future pandemic vaccines at least?
Yeah, I think it's an excellent question. From anyone wants to take it? Otherwise, I mean, I think Robin kind of gave an answer in that direction, that I guess it will still require the harmonization.
I can take a stab at this. Is
Yes,
that I'm Mark. I'm from Janssen Vaccines, and what we have noticed is that the majority of the countries do not require an ATT test to be conducted. There are a few countries that do have it in their regulations to perform an ATT test, with which we're currently still discussing, and there's no outcome yet whether the ATT in the end is needed. Unfortunately, there are still also, in addition, some countries that stick to their national regulations and do require, especially for new platforms, that the ATT test is being conducted for routine drug product release. So that's the current state. It's from our expectation, from our experience. The countries that do require an ATT test for routine drug product release, there are then discussions ongoing whether to conduct this particular test, let's say, endlessly, or to get to an agreement where after an X amount to be defined number of batches, the test could be discontinued, because then the data profile is sufficient to conclude that it's okay.
So thank you for that. Actually, that gives us some insight. But can you expect waivers from some of the NRAs?
Yes, I can expect. Yeah, I do expect waivers from some of the NRAs, definitely. Yeah.
Good.
But not all. That was kind of like the message. Not all of them.
Yeah. Okay.
So John, can I jump in on
Yes,
Mark?
please,
Mark, thank you very much for your response on that. That was very helpful. And Robin, to yours on the COVID. Health Canada would echo exactly what Robin said with regard to COVID vaccines. And in fact, it's come up a couple of times today that somehow in 2020 we stopped using the ATT. No, we started stopping requiring ATT back in the mid 2000s, in fact. And I would say that as a Health Canada rep, that I'm sort of somewhat disappointed it took us that long. Several of us within the agency thought that this was an irrelevant test when we joined the agency in the early 2000s. And I want to go back to Catherine's talk and say that, you know, this really dedicated effort by Klaus Kussler at the Paul Ehrlich Institute, and many of the elements of Catherine's talks were drawn from some of Klaus's earlier presentations, is this test became scientifically irrelevant, you know, many decades ago. And it's simply a reluctance of regulatory agencies to recognize that. In the chat, I want to direct ourselves to a question that came up in the chat. It was from a Russian colleague who was saying that the pharmacopoeia requires them to do that. And here is one of the major problems that many jurisdictions, just as in Europe, you know, it's the European pharmacopoeia that drives much of the quality control testing for good reason, but it's amending those pharmacopoeia, which is essential to prevent the continuation of the implementation of the ATT when it's been internationally recognized by WHO, EDQM, et cetera, many publications, as this scientifically irrelevant. So it's incumbent on individual pharmacopoeias to amend themselves through whatever process they have to remove that as a requirement for their jurisdiction. It's not always an easy process. I fully recognize that. I'm very fortunate that Canada is an observer at EDQM, so we actively participate in those discussions and have in many of the elements that Catherine's discussed. But I think EDQM is a model agency to show how regulators can reform and do so in a very coherent way. And it has the ability to, for example, remove the ATT from all of its monographs. That's not even feasible within WHO structure. They have a different system. It's a much, as Danyang has described, a more complex process. But it's incumbent on pharmacopoeia to stay current with science and to allow innovation in science. And ATT should have been removed back in the 1940s to be scientifically valid, or at least in the 70s and 80s. We shouldn't even have waited till the 90s. We knew this wasn't a relevant test a very long time ago. Over.
Thank you, Dean. And indeed, the question, in fact, in the chat is also a bit focusing on the recalculation of non-clinical data for rats and mice for the test dose. However, I guess this would be quite product specific. And I think the main element here, and please, everyone else jump in on it, but the main element here is to underline that the ATT remains an unspecific assay at the end of the day.
Shahan, just to go back to a comment Robin made, sorry to jump in, but is that she did talk about the requirement for the new COVID vaccines as requiring preclinical data. But again, that's a very focused set of tests looking at very specific things. And that is, of course, normal regulatory requirement for all new products. And that it provides us with useful preclinical information as a follow-up pieces, which is sort of the antithesis of what the ATT is. So yes, preclinical data is very important, but the ATT is not scientifically relevant. That's, I think, a key message. Over.
Indeed. Maybe another question. I think there's another question in the chat on the COVID-19 vaccine, but I think Mark has already answered it. It was basically going in the direction of the consistency approach, that if you cannot release the product without the ATT initially, then after you've generated data, that it can be removed. Maybe to switch a little bit to another topic, that was also one of the questions that have been handed in before the session has started, is on the adverse events following the vaccination, and how can this be tested, and how can the safety of the vaccine be ensured? Which other testing parameters need to be in place? That was one of the questions asked up front.
So I'll just hop in quickly with a statement, and really any post-licensure pharmacovigilance plans should take into consideration both the clinical aspects of the adverse events and any potential product impact as it's related to that. But I don't think that performing an ATT test at the time of release or in response to an adverse event in terms of a investigation of that event from a product point of view is going to really answer the question. And Dean's example of the heparin situation is exactly that. I think the types of product-related post-licensure pharmacovigilance follow-up would really look at per lot the specific features of the manufacturing process and whether there's any impact. So I don't think even in the post-licensure space with respect to pharmacovigilance that the ATT is the appropriate test to address the issues associated. So over.
No, indeed, and maybe another way to ask this is because if we take Canada or the US, where the test is no longer in place, have you seen an increase of adverse events since the time the test has been removed?
Hardly, no. It is in fact entirely scientifically irrelevant to our process, and Robin described that very well. I think Mark could comment similarly, and please do. The rigor that's applied to manufacturing of biologics now is immense. It's a massively costly and rigorous process. So people should understand that. The notion that this ancient test somehow provides any measure of security is as acknowledged by WHO, who itself was slightly late to this game. And again, that's Karl-Heinz Bruheck who brought that to them and worked collaboratively with Paul Ehrlich to ensure that that was passed at WHO. It has no scientific basis. Nobody should be, just because we can do a test doesn't mean it's relevant to purpose, and that the ATT is a perfect example of that. And I want to also just talk about another element. The scientific rigor applied to manufacture now is such that having the appropriate instrumentation to do those tests becomes very difficult for smaller agencies smaller agencies to even afford to keep up with. And this goes to a piece from Thierry, is that one thing that I think can drive harmonization is reliance between and collaboration between agencies. So again, the EU is an excellent example of many small or middling sized countries who have banded together to form a regulatory network, and they combine their resources to do that, and they do their lot release by and large in a collaborative process as well for vaccines and other products. And that's an excellent model for on a worldwide basis. Many smaller countries may not be able to keep up with the sophistication of manufacturing that would provide robust confirmatory testing, but on a more focused basis. Having testing going on for the same lot in multiple jurisdictions is actually a giant waste of our limited regulatory resources. But coordinating that testing in larger blocks, ensuring that the laboratories doing those testing have themselves an appropriate quality control system, can afford the appropriate technology and do so, and do that on a regional basis is a much, much better way to go. And that will actually provide better assurance. And a lot should only be tested by the manufacturer and a competent authority to do so, and a well-resourced competent authority with the proper quality controls in place. And we could coordinate between regulatory agencies to do that. So this is something that we frankly owe industry to maintain, to work towards that global effort that has been talked about. And it's something that all agencies should be considering very carefully and looking beyond their national borders and their pharmacopoeia.
And just to add on to this point, it's a very good comment from Dean. The ATT test is known to provide variable results, and basically it's not reliable to a certain extent. The QC department of the world has started to adjust the method so it would fit the results, so you would simply get a pass. And to a certain extent, I do not think that anybody knows what is the end result of that test anymore. What does it mean? So for the foundation, it's extremely important that we don't set up the developing country vaccine manufacturer for failure, but just generating all those tests and ending up with a portfolio of tests that with no meaning. We want them to have access to new technology. We want to have access to the latest pharmacopoeia in terms of range of tests. And eliminating ATT is just the tip of the iceberg. So I really like what Thierry said at the beginning of the talk in terms of the new R, the rely, reaching out. So basically we need that standardization of methods around the world. But I think now it's pretty obvious we need a standardization of removal of the methods. Because again, ATT is just one example of it.
Indeed, it's an excellent point. And also Thierry underlined one. Please go ahead.
Yeah, I just want to add one point here. Being Indian manufacturers, we are still following ATT test as a requirement, because in our pharmacopoeia, we have a general chapter which says that if tested by NRA, National Control Laboratory, it should pass the test. So as a manufacturer, we think that first we should see the result before our national laboratory sees the result. So we are still following that as a requirement. Unless our regulatory doesn't have that point, we still follow that.
And I think this one fits quite well with the question that Lisa Plitnik has posted in the chat. So we'll just read it. So it's clear from the panelists that there's a consensus on the ATT should be deleted. But what is the rationale for the countries for keeping the test in place? And then speaking from her own experience, we've been providing the same valid rationale for deletion for quite some time and had made significant progress. But what has changed that will now convince them to delete it. So
if I can jump in on that, Shahad, there is no rationale to maintain the test. Any pharmacopoeia or any agency that holds on to it, and I'll say this very bluntly because I would accuse ourselves of the same things. We as Health Canada should have engaged in this discussion in the late 1900s rather than the early 2000s. There hasn't been a rationale for this test for a long time. And it's simply a matter, there is a tendency for for people, and regulators being people, to hold on to things they know, whether it's an ATT test, whether it's an animal potency test, even when it's very clear there are alternatives that can do a far better job, far more sensitively, far less expensive, far quicker, etc. People tend to go with what they know. So the notion that if you can't do testing, but you can manage to do an ATT test, it's not a rationale to continue doing so. Just as Philippe was mentioning, working in a harmonized structure, in a regional structure, as the African Medicines Agency is attempting to set up, for example, for that entire region, and having sophisticated technologies that can advance product development is the way to go, and modifying the pharmacopoeia requirements is essential. That has to be expunged, just as the European Pharmacopoeia has done. Go ahead, Rob.
So I'm sorry, Dean and Shahjahan, this is Gautam. I don't want to jump in if somebody else is ahead of me.
Yeah, and then I think after you, maybe Robin can also comment.
Yeah, no, that I'd love to hear Robin's comments. But this whole time, actually, this question was right on the spot, and I was hoping, or I was wondering if we could hear from any representative present, or if present from one of the countries that that do not accept waiver or have not deleted it. I have heard the argument, at least from one country, that we do it. We do a slightly different test, or by we I mean their national regulatory agency. I think we apply it, somewhat differently for maybe different products, but at least the test is adapted for, to fit the purpose. But I do not exactly know.
Gautam, I fully understand your point, but I think therefore we will have the second round table.
Oh yeah, I'm sorry. Okay, yes, you're right.
Yeah,
Thanks for reminding me. Yeah.
So sorry for being splitting things, but I think
that's good. Yeah, thanks.
Yeah.
Robin, maybe I can give the word to you because you wanted to comment.
Yeah, I just wanted to follow up on one of the things that Lena had said, and I totally understand that if your regulatory authorities are doing the test, that you also need to do the test so that there's not discrepant results out there, or that you're not put in a position to have to justify something. But is there any dialogue about even the regulatory authorities no longer doing the test? Or is there some flexibility there? And I know this is kind of also for the next panel in terms of for countries that are still involved in this, but since you brought it up, I was just interested in where the discussion is.
Yeah, so Lisa, could you jump in directly? Otherwise, I mean, I can try to answer the question for
sure,
GSK.
Thanks. I think.
No, otherwise I can try to answer the question for GSK. So GSK at least is in this direct conversation with national regulatory authorities that still require the ATT, and try to convince at least that if the test is required, then it's only, as Dean has explained also, that it's only then done once and the data is at least shared and not repeatedly done. However, as we also know, the ATT is also not a harmonized method at the end of the day. So it still, I think, remains a difficult topic.
So John, if I can also suggest that another way to move this issue forward, to put it on an international dialogue, is to go to the ICMRA. WHO is, of course, has a broader reach, and has done its work on this by declaring it a non-scientifically valid methodology, not relevant for purpose. I think bringing that up at ICMRA, which is an international body, again, of regulatory agencies, it doesn't have the broad reach of WHO at this time. It's at least 20 or 30 agencies, but many of the countries sitting at this table today are represented there. And having that discussion at that level, just again to go over the irrelevance of this test and its impedance of lot release and undue regulatory burden is a good forum perhaps to consider industry bringing that to. And there has at least been one joint industry ICMRA meeting recently under the COVID banner. Perhaps this is another opportunity to raise this type of testing and its deletion and further harmonization.
No, definitely. And I think you cannot advocate enough and and communicate enough about the obsolescence of this assay. Huh? So I think that's a great follow-up action of today. Another question that was asked up front was regarding the benefit that it would bring to remove the test. And Jan Ling, you already mentioned the time, so the time to release the batches, but maybe also to ask to Mark as a manufacturer, where do you see other advantages of removing the test?
Well, first and foremost, as it was pointed out by the speakers, I mean, animal suffering should be refrained and should not, the animal should not endure any suffering. So the animal's welfare is one very important point, at least to us within Janssen. Secondly, all these tests, they need to be developed. That actually costs time. Although it's a compendial test, you have to always perform the analysis. So it's not only the testing itself, right? Also the prep work that you need to do there. From a release perspective, for the lot release perspective, dependent on the country you go to, as mentioned, there are different ATT tests. So you cannot go to one country where you conduct the ATT and then say, well, look, this test can be used for any other country. There are a number of versions, let me phrase it like that, of the ATT test. So already that combined from a manufacturing perspective requires all kind of pre-test in advance for a certain nation. Then you have to conduct that as part of your release process. Then the regulatory entity is going to repeat that test again to confirm the data that you have. All of this basically takes a lot of time and requires a lot of animals with hardly any added value, as pointed out by several speakers today.
Thank you a lot, Mark. And so we get more and more discussions in the chat, which is great.
Can I just ask a quick follow-up?
Yes, go ahead.
Mark, have you seen any relief given that Europe and US and many countries are starting to decrease the reliance, or are you still, there really hasn't been any change given that you have to do it anyway?
I think this is a great question, Robin, and what we actually I tend to think that there is kind of like a snowball effect ongoing, where since the larger institutions have been saying remove it, and they're in the process of removing it, and I think that that's the important point, is when the larger agencies are going to remove the test and any reference of the test in any of their guidelines. The somewhat, I don't want to be unpolite here, but the somewhat smaller countries and the national authorities, they will follow that. I mean, we have far fewer of these type of discussions now than that we had in the past. And hopefully this, let's say, progressive movement will continue once from the agencies the test has been removed from the guidance, to expedite the removal for other countries. And also that will be, I think, a stronger position to those last countries that do continuously insist to execute the ATT test. Because then they would be the very last, and they have to come up with very good explanations why they would like to continue and have this test being performed, although the rest of the world is saying, you know, we don't need to have that test anymore. And I think that's the collective and the reliance that we have been talking about here, that is needed in order to get this test off the table for the entire world.
Thank you a lot, Mark. And so we only have a few minutes left, so maybe a short closure statement. Philippe, can I ask you to start? What would be your statement, your one sentence as a message to organization and people that still rely on the ATT?
Yes. So the importance to people to connect, really exchange that information in terms of removal. And that's why we have that expression. So DCVM for development country vaccine manufacturer, but it's the N at the end. DCVMN, it's a development vaccine country manufacturer network. And that's how we're going to be able to prevail together and standardize.
Thank you, Philippe. Lina, can I give the word next to you?
We as manufacturers, we also want this ATT deletion, but with a thought that consistency to be demonstrated on the biological product. However, this is only for initial batches like clinical batches and few batches till the demonstration of consistency. And with a clause that if there is any change in the manufacturing process which may have an impact on the safety of the product, that also has to be evaluated, and it has to be proved for the product is safe in terms of ATT.
Yeah, so they would still be under the consistency approach umbrella and not a complete deletion. But that's quite clear. Yan Ling, can I ask you for your final sentence?
Yes, thank you. Yes, and WHO has its clear position on to remove this ATT test from the lot release testing. The ECBS gave the scientific advisory recommendation to the national regulatory authorities. This should be implemented, or this can be implemented by the national regulatory authority, and to promote the implementation of the ECBS recommendation is very important, whether we through the bilateral dialogue with the regulatory authorities in the country or through the ICMRA, and we can pass the message to the country and have a dialogue with the country and to understand their concern and to explain the scientific basis of remove these tests, by WHO. I think we will do our best and to dialogue with the member state and to implement the ECBS recommendations into their regulatory practices. Thank you.
Okay. Thank you very much. I think we are already running a bit over time. So Robin, Dean, Mark, I hope it's okay for you that we take a break now before we come back. But I think the overall message is quite clear. We need to have more harmonization on the deletion of the test. As long as there's a loophole, it will still remain in place one way or the other. Thank you very much for the very good discussion, and also for the audience to ask the question directly and speaking up. So we now go for a short break, and then I think we're back in 20 minutes.
Thank you.
Thank you. Bye-bye.
Thank you. Bye.
Thank you.
Thank you.
Sorry, we are back in 5. We keep the session as short as possible. So thank you very much, and we start soon.
You can take the lead, yours.
Okay, thank you, Laura, and thanks to the previous speakers who really set the scene already. Very brief as an introduction, I am Joris van der Putte, the president of the International Alliance for Biological Standardization, and going immediately to the focus of the discussion in 2019, with SHI, with Laura, and with the VAC2VAC consortium, and with the Thai authorities, we organized a first 3 hours meeting, non-animal testing for batch release. And I must say, we had a lot of positive input and impact, at least as I understand it, from our Asian colleagues. So that being said, there is a lot happening already, and I share the concerns of Thierry, but there is already a lot happening, but as human beings, sometimes it needs time. So today we have an extraordinary panel, and they each have respectively a time slot accorded, and I will try to keep to the time slot. So we start with our Japanese colleagues, Tao Mizukama, and Yoshihisa Shirazaki. So I leave it first to you, and then it will be followed by our Chinese colleagues, probably the Russian colleagues, the Indian colleagues, Indonesia, and South Korea. So I believe it is Yoshisha who starts. The floor is yours.
Yep, I think so. This is just speaking. Dr. Mizukami will start first.
Okay, thank you.
Okay, I start about answer the question. So in Japan, so inactivated vaccines need to pass the ATT for both manufacturers testing and independent rotary testing by National Institute of Infectious Disease, my institutions. For live attenuated vaccines, the independent rotary testing for ATT by NIAID was not implemented, so that only manufacturers testing is required. So every year, an average of 250 lots of vaccines are released, so more than 500 ATT are estimated to be performed every year, resulting in the use of 1,000 guinea pig for ATT. However, since 2008, we started to consider an exemption of ATT in vaccine lotteries for recombinant vaccines, as they are not derived from live viruses and bacteria, and lotteries protocol for vaccines will be introduced by 2011. So we assessed the previous historical ATT data of the recombinant hepatitis B vaccines and finally decided to introduce a waiver rule in the hepatitis B vaccine lotteries. So given that it passed ATT in the previous 20 consecutive lotteries test, so the Amendment Act was first enforced in 2013. So since then, we have evaluated the historical data for many vaccines that have met our criteria. So already tested for more than 5 years or 50 lots and low retest rates and no report of typical adverse event because of the vaccine quality. So we also implemented the waiver rule for the pneumococcal conjugated vaccine in 2016 and influenza HA vaccine in 2017. The Japanese encephalitis vaccine in 2017 and Haemophilus influenza type B vaccine in 2018. So all Amendment Act were enforced in 2020, so which result in the complete waiver of 70 to 8% of ATTs for vaccine lotteries. So we are still evaluating the historical data of some recombinant vaccines such as human papillomavirus vaccines and the conjugate vaccines to introduce same waiver rules. So regarding live attenuated vaccines, so we correct the in-house test result of all manufacturers and found that there are no batches that fail by the ATT, so which provide evidence that ATT is no longer needed to determine the safety and consistency of the live attenuated vaccine. So after the future scientific committee meeting in our institution and the crosstalk with manufacturers, we will be able to waive the ATT requirement for all live vaccines by early 2022. So with regard to plasma-derived medicinal blood products, so more than 400 blood products were released every year. And similar to vaccines, most of the blood products need to pass ATT by manufacturers' in-house testing. So independent lot release testing by our institution for ATT has already been waived since 2005. So 400 lot of ATT has been done in Japan by manufacturers. So since 2020, we correct the in-house test result of all manufacturers and found that no batches failed by the ATT. So we also evaluate the historical ATT data and found that body weight reduction and the typical pathological changes has not been observed in the blood product for nearly 20 years. So this provides strong evidence that ATT is no longer needed to determine the safety and consistency of blood products. So after the scientific committee meeting in our institution and crosstalk with manufacturers, we finally decide to delete ATT requirement for all blood products. The amending act will be enforced in this next month, late 2021, and this will result in 100% ATT waiver for current and future blood products. So we are now planning to design the roadmap to delete ATT requirements for the rest of the vaccine within a few years.
Thank you very much. Can I hand over to Dr. Yoshisa? So
Okay.
you limit, if possible, to 3 minutes, and
Okay.
if possible to the dialogue industry and regulators. Sorry to be a little bit directive because we have a lot of other speakers to follow. Thank you for your
Okay. Okay. Hello, I'm Yoshisa Shirasaki, a member of Vaccine Division, EFPIA Japan, and Japan Vaccine Industry Consortium. I'm from CMC-RA department at GSK Japan. So regarding first question, so what is the current requirement of ATT in your country? So in Japan, ATT is defined as a general test in MRBP. MRBP is minimum requirement for biological products, which is an official compendium for vaccine and blood products. So after new vaccine submission, NDA in Japan, NIID, National Institute of Infectious Disease, may request to submit ATT data for 3 lots during pre-approval testing process and to set ATT as a specification item for QC release test in the most case, even if company does not set ATT as a specification item. So in vaccine approval timing, so a monograph for the product must be listed in MRBP, where ATT would be a part of specification item in the most case so far. So on the other hand, ATT for all blood products will be removed from MRBP monographs soon. So ATT for some vaccine products confirmed the process and product consistency by NIID can be omitted, as explained Dr. Miskami. So for second question, so how manufacturers and regulatory authorities communicate and discuss about the deletion of ATT from the pharmacopoeia or local regulation. So we formed Japan Vaccine Industry Consortium, which is consists of EFPIA Japan, Pharma Japan, Japanese Pharmaceutical Manufacturing Association, and Japan Association of Vaccine Industry. So Japan Vaccine Industry Consortium are discussing with Japanese authorities, that is MHLW, Ministry of Health and Labor Welfare, and NIID, and PMDA, Pharmaceuticals and Medical Device Agency, about deletion of ATT from MRBP general test through having several workshops so far at least 4 years. So Japan Vaccine Industry Consortium considers that ATT is no longer needed, since GMP manufacturing process has been implemented for several decades in Japan, and process product consistency are confirmed by raw material control, process validation, in-process testing and QC release testing, etc. And this is our comment. And the next question, so what are current obstacle challenge for your country to delete complete ATT? So regarding this question, so from industry side, NIID considers that ATT results for each lot is useful to monitor the consistency of manufacturing process and product. Therefore, it seems like that NIID is reluctant to delete ATT from MRBP soon. So on the other hand, MHLW considers that the regulation for vaccine products, including ATT, should be aligned to the international strategy. However, MHLW explains that they will take some time to investigate the justification for deletion of ATT from MRBP. So, according to Japan Pharmaceutical Industry Vision 2021, issued by MHLW on 13th September this year, the authorities will proceed international harmonization of regulation for vaccine products and consider ways to expedite and simplify procedures, including the abolition of animal testing, that is ATT. That's it from Japan vaccine industry side. Thank you for your attention.
Thank you very much for your effort and for this two very explanatory comments on the situation in Japan. May I remind all the participants to make their questions and comments on the chat. I do not see any, and I propose to keep the questions for the end. So now we pass over to our Chinese colleagues, and we have a couple of representatives, Dr. James Oy, Dr. Yingying Zhu, Dr. Xiangtang Li, and Dr. Jack Xi, and Professor Yang. And according to our preparatory meeting, it's Dr. Jack Xi who will start. Is that correct? Jack, the floor is yours.
Thank you. So I can start first. Our industry company members are from ADIPAC members. ADIPAC is industries.
May I just ask a question also to all the panel members, not to forget the key issue of dialogue between industry and the regulators in your respective countries. Thank you. Over.
Sure, thank you. So as I just mentioned, all our members are from ADIPAC Industry Association. This association consists of 42 multinational companies. All these companies have R&D capacity in China. We had a lot of interactions with health authority of the ATT. I will first talk about the current requirement on ATT, then Xiantang from Pfizer will give a summary of our effort to delete the ATT in China, and Yingying will cover our estimate evaluation on the current obstacles. So the current requirement in China, according to the China Pharmacopoeia, is required for the both all biologics and vaccines. And for biologics, it's required for the registration and the product batch release after marketing. And for most of biologics, only need mice test for the ATT. For vaccine, at least for, if not all, at least for some vaccines, for example, like a COVID-19 vaccine and several other vaccines, it requires ATT at the CTA stage, or sometimes before the first in human stage. And also for these vaccines, according to the China CDE and FDC's requirements, it requires both mice and the guinea pig test. And in the most recent version of China Pharmacopoeia, it's a 2020 version. After the many years effort by our ADIPAC companies, a statement was added to this requirement for the biologics. It says that if the quality of the product, the attributes of the product itself not suitable ATT can be waived based on the approved solid rationale and risk assessment. This is a big progress. However, based on our experience, it's hard to justify and hard to get waived by the communication with China Pharmacopoeia. I think I can stop here. Maybe Xiantang can address the next part.
Thank you, Jack.
Yes, I think just as Jack mentioned, we DS already, with the already PAC company worked jointly together in past several years, try to pursue the Chinese pharmaceutical committee, the pharmacopoeia committee to delete this ATT. So especially in those 2018 to 2019, those 2 years, just before the current China pharmacopoeia publication in 2020, we tried to very hard to pursue them to delete the ATT. And we shared them with publication, including from our own member. So, for example, Jack also published a paper. So why there's ATT's no value, use the specific his case sample when he was at Roche. And also we shared the EFPIA, the white paper in 2017, and we shared all of those WHO related documents, the guideline to with CDE, China Pharmacopoeia Committee, and also National Institute Food and Drug Control. Also, in particular, the IQ to Save team, the IPAD, the already PAC non-clinical working group, we plan to jointly write a white paper to the Chinese Pharmacopoeia Committee. Finally, there's the To Save IQ had a similar the white paper already, so we just modify, revised slightly, and submitted it to the China Pharmacopoeia Committee. So essentially, or in summary, I think the already PAC and many of those pharmaceutical company in China and a local China pharma company devoted a great effort to try to pursue the Chinese regulatory authority to delete ATT from 2020 China Pharmacopoeia. Just like Jack said, they did not really completely delete the requirement, but they made those specific statements based on the CMC data and other rationale and to risk assessment can request for waive ATT on case by case. But again, like Jack said, probably difficult to justify because there's no specific or concrete criteria. So there will be some kind of gray area. So maybe not easy to justify to waive there. So basically that's the summary, whether we have the effort in past several years. Probably we will continue to work. Hopefully this ATT will be completely deleted in 2025, Chinese pharmacopoeia version then.
Okay, thank you very much. So,
hi.
Yeah,
okay. So this is Ying. So I will share some of our discussion as what the obstacles and the challenges. I guess a lot of these are actually just our speculation because we think China actually right now is in a very unique situation. As probably all of you are aware, China has set a big goal to transform their healthcare system. So we have seen a very fast growing and very dynamic period of the time. A lot of evolving regulatory policies, as we can see, hundreds of new guidelines have published over past few years. And we also have seen a lot of new companies emerges. So this reform did lead to accelerating the clinical trials and the new product approvals, many positive impact, but we also see heterogeneous growth in some areas, like some big farms actually with very advanced technologies and plenty of resources, but some small domestic companies may have limited resources, inconsistent safety control process, or GLP compliances. So it's our speculation that the China regulatory agency probably try to strike a balance to solve this heterogeneous situation. As Jack and Xiantang mentioned, they did have some progress to add some justification in the 2020 version of China Pharmacopoeia to allow some flexibility. But on the other hand, they still take a very conservative approach to keep the majority of the regulation as it was before. So basically, it's our RDPAC's intention to keep these, interactions with the agencies and have more dialogues, try to support and push China to remove the ATT requirement, hopefully in the near future, next version.
Okay, thank you very much.
Mm-hmm.
Is there any other remark? I think I heard someone now. I see no questions from the chat. So I just have been informed that Professor Yang is not joining. So I propose that we pass over to our Russian colleagues now. Sorry.
Thank you for the opportunity. And the abnormal toxicity test is included both in the 14th edition of the State Pharmacopoeia of the Russian Federation and volume 1 of the Pharmacopoeia of the Eurasian Economic Union. And this test is included in such monographs of Russian Pharmacopoeia as active pharmaceutical ingredients, parenterals, recombinant DNA medicinal products, allergens, bacteriophages, vaccines and toxoids, immunobiologicals, human immunoglobulins. It should be noted that the ATT test conditions differ in the Russian Pharmacopoeia and test and the withdrawn European Pharmacopoeia test. The test described in the European Pharmacopoeia was less stringent than those described in the Russian Pharmacopoeia or USP. The injection time in the European Pharmacopoeia was 15-30 seconds in contrast to 5 seconds in the Russian Pharmacopoeia, which produced divergent or ambiguous results. It has also been demonstrated by experiments that a long injection time didn't allow for detection of toxic effects of such medicinal products as antibiotics of natural origin. At the same time, the question of ATT deletion for a range of medicinal products is currently on the agenda for both Russian and Eurasian Pharmacopoeia. Based on the test results obtained during expert evaluation, scientific recommendation on ATT removal were published for the following groups of products: water and normal saline for injection in packages of less than 50 milliliters, orphan medicinal products, local anesthetics, and discussion are in progress for 2 more groups: MAPS for subcutaneous administration, insulin and its analogs. Presently, the importance of these tests for vaccines is under thorough examination. 114 vaccines are licensed in Russia. In 2020, there were 54 vaccines available in the Russian market, and 48 of them had to be checked for abnormal toxicity. No biologicals have been found problematic in terms of abnormal toxicity since 2012. Nevertheless, in order to delete the test, the following conditions ensuring the safety of manufacturing should be fulfilled: the manufacturer's compliance with GMP rules, full validation of processes, adequate control of the product's quality and formulation stability, including APIs procurement from well-established certified suppliers. At the moment, the decision on ATT withdrawal from the product specification is taken on a case-by-case basis for every medicinal product. But for many products, we apply removal of this text from the routine batch release. The main argument in favor of keeping ATT in the Russian pharmacopoeia quality control system is that it detects medicinal products not compliant with pharmacopoeia requirements in terms of toxicity. The data updated by our scientific center and Roszdravnadzor over the past 5 years shows that widely used cephalosporin antibiotics such as ceftriaxone and cefepime have been repeatedly found substandard in terms of this parameter, which makes it impossible to delete the test from specifications for antibiotic of natural origin. To sum up, I would like to emphasize that Russia is moving towards replacement of animal testing with in vitro tests to the greatest extent possible, but our decisions should be based on the empirical results and scientific journals. Thank you for your attention.
Thank you very much. Now I hand over to Dr. Professor Viktor Alexandrovich.
Sorry, but our colleagues now are in Siberia and can't connect with us.
And the same for Dr. Elena Sakanyan.
The same for Dr. Elena Sakanyan.
Okay. Thank you very much, Dr. Trapkova, for your excellent presentation and your view on the future about the ATT. So now we hand over to our Indian colleagues, and it seems that our friend and colleague, Dr. Sunil, will start the presentations on India. I know you sent already a message on the chat that in India, the ATT is already abandoned. That's very good news. Sunil, the floor is yours.
Yeah, thank you, Joris. And hello, everybody. And it's a pleasure for me to inform that abnormal toxicity test has been deleted from human vaccine monographs in Indian Pharmacopoeia in its amendment in July 2020. And going back, this entire scenario started way back in 2015, where we as manufacturers got in touch with our NRA, with Indian Pharmacopoeia Commission, put forth our request for deletion of abnormal toxicity test, and also to have waivers for certain other tests. And IPC was quite considerate enough to take our request into consideration. And we had a series of meetings with National Regulatory Authority, the joint meetings with Indian Pharmacopoeia Commission and the manufacturers. And the efforts paid success. And in the year 2018, when the new edition of Pharmacopoeia was launched, then abnormal toxicity was waived off conditionally. That is, the test, the manufacturer should demonstrate the consistency of production to the satisfaction of NRA. And if the test is to be performed by NCL, it should pass. So it was considered as a waiver. And we as manufacturers, we are around 20 manufacturers in the country. We were still not satisfied with this condition. So we further requested Indian Pharmacopoeia Commission to take our request further for discussion. And based on the submission of substantial amount of data, substantial amount of monographs, and the pharmacopoeial discussions took worldwide, and we were able to convince the Indian Pharmacopoeia Commission. And in the year 2020, in July, complete deletion of abnormal toxicity test was announced in the month of July for the human vaccine monographs. So which waived off the condition like consistency establishment or testing by NCL. So that gave us a lot of relief. Not only abnormal toxicity test, we have taken up many other issues also. And it is due support of Indian Pharmacopoeia Commission that we are able to go ahead. And now we are committed to follow the principles of 3 Rs. And we are going very well ahead in this direction, and hope in near future we announce some more of such type of deletions of tests. So may I request Dr. Jayaprakash, he would like to add anything?
Thank you, Dr. Goyal. Am I audible?
Yes, please.
We hear you very well.
Okay. So at the outset, I thank the organizers for inviting us for the at the outset, I express my gratitude to the organizers for inviting me to this important virtual event related to the accelerating global deletion of the abnormal toxicity test, planning the common next step. We agree with Dr. Sunil Goyal, and IPC is fully committed to adhere to the principles of 3 Rs, and we come out with the Indian Pharmacopoeia every 4 years with its addenda or addendum in between, and the general chapter of Indian Pharmacopoeia Commission of the in Indian Pharmacopoeia 2.2.1, it specifically gives the description of the abnormal toxicity test, and the general notices of Indian Pharmacopoeia, we have included a specific clause for prevention of cruelty to animals act 1960 and adoption of the CPC SCA guidelines. I would also like to add that ATT had been removed from the human vaccines of IP. So via the amendment list number 6, and we keep on bringing the amendment in Indian Pharmacopoeia, keeping in view the feasibility of the test as well as the alternative methods in the pharmacopoeia. As Dr. Sunil Goyal had already informed, that the abnormal toxicity test had been omitted for the routine lot release once the consistency in the production had been established to the satisfaction of the National Regulatory Authority, and when the good manufacturing practices are in place, and each lot should comply with the test for abnormal toxicity when tested by the National Control Laboratory. And we have the Central Drugs Laboratory as one of the National Control Laboratories at Kasoli, as well as the National Institute of Biologicals. These are the two important national regulatory and national control laboratories to test independently the vaccines for human use. As regards the TABST test in the veterinary vaccine monographs is concerned, we are also considering it, and during the workshop of the TABST and LABST, which was organized by Human Society International on 11th of February 2021, we had asked for the retrospective data of the consecutive batches from different manufacturers and submission of the clear proposal so that we can put it up to our veterinary expert committee for consideration. So we are taking all the steps for the quality control methods as regards human vaccines as well as the veterinary vaccines standards are concerned, and we are sensitive to the uh, request of the manufacturers, and we also look into the feasibility aspect where the standards can be whether they can be fully applied, whether they are feasible or not. So once that is all practically feasible, then only we bring out the changes in the Indian pharmacopoeia. And over the past few years, there have been a lot of regulatory changes in Indian pharmacopoeia, and not only in the area of these specific tests, but also for adoption of the alternative methods as and when they apply as per the text of the Indian pharmacopoeia. We try to adopt the concept of the harmonization while implementing and bringing out the standards in Indian pharmacopoeia. There is a general chapter on vaccines, the different type of the tests which are required to be carried out for human vaccines, and that has to be read in conjunction with the specific monograph set out in Indian pharmacopoeia. So this is all what we have done, and we are keen to further strengthen the science-based monographs in the interest of the animal health and also in the interest of public health. Thank you very much.
Thank you, Dr. Jayaprakash. I would just like to add, Joris, in addition to deletion of ATT from Indian pharmacopoeia, but Serum Institute being an exporting country, we still have to perform the abnormal toxicity test based on the importer's requirements. There are certain countries for which we have to specifically perform this test. So I would put this point for further discussion for implementation of deletion of abnormal toxicity test globally. And we are open for any questions for discussion if you have. Thank you.
I agree with Dr. Sunil Goyal. Thank you.
Thank you very much, Dr. Prakash, and thank you very much, Sunil, for taking up this question and remark again. We already discussed in Bangkok in December 2019, and it's a very good evolution to go forward with that for global harmonization indeed. So last but not least, now we have our, it's not last, first last but not least is our Indonesian colleagues. So we have Dr. Unik Suwarni and Dr. Amrullah Aninditio. So it seems that Dr. Amrullah will open the discussion. I hand over to you, Dr. Amrullah.
Okay, thank you. Thank you for this opportunity. We carry out intensive communication regarding this matter. Harmonization is highly ordered between biopharma as an industry with Indonesian National Authority, especially from Quality Control Laboratory in NRA, and of course of the elimination of this abnormal toxicity will reduce our testing time for vaccine release, and also our commitment as industry regarding to the 3R agenda. One of the things that we're still discussing is wherever is sufficient to cover it with the sterility test, endotoxin test, or pyrogen. And then whether this ATT elimination will apply completely or will be carried out gradually in certain vaccine. Maybe it's from us. I can offer to Dr. Enik.
Please, Dr. Enik.
Thank you very much, Joris and Dr. Amrul. I'm from FDA, Indonesian FDA. The SWA abnormal toxicity tests have been carried out for safety control to all vaccine products, such as TTP, measles, hepatitis B, pentabio, COVID-19, and etc. Other safety testing performed in Indonesia are endotoxin and sterility tests. Based on the development of science, some organizations like WHO, Expert Committee on Biological Standard ACBS, European Director for the Quality of Medicine, Edqm, have to delete ATT, like in the video early and has been mentioned by the previous speaker. Indonesian FDA has studied on abnormal toxicity tests from some sources, such as British Pharmacopoeia and European Pharmacopoeia. The studies showed that ATT doesn't seem to be relevant in monitoring dangerous effects of vaccine products. Additionally, ATT use animal laboratory, which may not be according with 3R, replacement, reduction, refinement, it's for animal welfare. Currently, Indonesian FDA has proposed that ATT is eliminated from Indonesian pharmacopoeia. This is from my point of view as a regulator. Thank you very much.
Thank you extremely much, Dr. Enik. And thank you to the two Indonesian colleagues for clarifying also this wonderful interaction between industry and authorities. And now, last but not least, our South Korean colleague, Dr. Kyung Yin Jung. Please,
yes.
Dr. Kyung, the floor is yours.
Yes, thank you for introducing me. Hello, everyone. My name is Kyung-jin Jung. I'm a research scientist working at Korean Institute of Toxicology in South Korea. I'm glad to share the Korean status of ATT regulation as being an industry representative in this workshop. Thanks to the organizers. In South Korea, ATT regulation has not been removed yet from most biologics, which are subject to the regulation of standardization and evaluation. According to Korean regulation for standardization and evaluation for biologics, which is related with the Korean Pharmacopoeia, but ATT can be waived when there are manufacturing data approved by an advisory person in charge of quality control under the condition where the lot was produced in compliance with the quality control and without external contamination in the manufacturing process. Based on what Korean ministry said, excluding national lot release material, ATT is still being requested by regulatory authorities for approval of biological products. And still many industries are still following on it. Korean ministry said there has been no waiver case reported for the past 5 years. And they also said that they are preparing for revisions of regulation of ATT. In the meanwhile, since 2019 in Korea, regulatory authorities amended the official deletion of ATT for the national lot release materials, which of quality was monitored by regulators. In this context, recent 4 COVID-19 vaccines such as Moderna, Pfizer, Janssen, and AstraZeneca were not requested for additional ATT testing because regulatory authorities granted national lot release material to those 4 vaccines after final their quality check. That's all I answered.
Okay. Thank you very much, Dr. Kuhn. Now we have time for answering questions. There are quite a lot of questions for Dr. Trapkova. I see that Dr. Trapkova is willing to answer the different questions, so I read them. The first one from Suzanne Osborne is the question, can we apply Russia to exclude ATT from specification for genetic monoclonal antibodies subcutaneously less 3 milliliters volume therapeutic dose? A second question from Dr. Gautam Sunyal. How does ATT testing solve problems with substandard antibiotics or other products? Will they be approved if they simply pass the ATT? And then a third, and the other question is to Dr. Prakash. First, up to you, Dr. Trapkova.
Thank you for your questions. And for first questions, yes, of course, you can give an application to Russian Federation, and I hope that after examination, the results will be positive. This is my answer on the first question. The second question, how ATT test can solve the problem of substandard antibiotics? I can say that this test can't solve this problem of substandard antibiotics. The problem of substandard antibiotics may be solved by manufacturing with GMP rules, but ATT test can prevent the presence of substandard antibiotics in the Russian market. Thank you.
Okay, thank you very much. There is a question to Dr. Prakash, but it seems he answered already about the toxicity test omitted for routine lot release. Please, Dr. Prakash, we have a little bit time. The question was, is it removed entirely from the pharmacopoeia or is it just waived if a certain set of data are provided? And if data are required, what are those data that are needed? Is, for example, ATT data still required as part of those data? Dr. Prakash, if you are there.
Yes, yes. So let me respond to this question. So the vaccines general requirement in Indian pharmacopoeia specifies about the abnormal toxicity test. Appendix, the number is 2.2.1, and it specifies, unless otherwise stated, all vaccines comply with the test for abnormal toxicity method B, in vaccines containing phenol as the preservative, the test in mice may be inappropriate, and the abnormal toxicity test may be omitted for routine lot release. Once the consistency in production has been well established to the satisfaction of the national regulatory authority, and when good manufacturing practices are in place, each lot, if tested by the national control laboratory, should pass the test for the abnormal toxicity. So this is the specification set out in the Indian Pharmacopoeia. So one has to comply with all the text, the methods, and each of the clause, including the general notices of the Indian Pharmacopoeia, while properly interpreting the pharmacopoeial monograph.
Thank you very much, Dr. Prakash. I have a general question from Dr. Gautam Sanyal, who unfortunately has to leave, but I read it. It's a very important one, in my opinion, at least. So the discussion that happened in the previous session and in this one point to the need of concerted global harmonization, regulatory harmonization effort. So his question is it possible to have WHO take the lead in organizing such an effort? I don't know whether the WHO representative is still there. I do not hear, so this is a question we can keep in mind.
This is Gautam. I'm still here, and I'll.
Okay, good.
Yeah. So I thought about this for a long time. I mean, this is I'd say this is probably about the lowest hanging fruit. But there are obviously other tests too, and COVID, the pandemic, has really shown us how important it is to have global harmonization. I was really thrilled to hear from the South Korean representative just a moment ago that for those 4 vaccines they did not require, because if you're looking at the background with which these vaccines have been prepared, or many of the vaccines are being prepared these days, 1900 test which did not have any other tool to really identify anything practically at a molecular level is obsolete. I mean, it's a given, we keep talking about it, but so I think international harmonization, this is something that can set a really good precedent for others to follow, for other such tests to to follow, and I really hope that WHO could be in a position to take a lead, at least to organize the discussion, and maybe a workshop or something. Again, this workshop is fantastic, but a lead coming from WHO may set things or push things forward a little bit faster. So I'll stop and let you guys discuss this further, and maybe take it up later too.
Can I jump in and have some comment on that?
Yeah, please.
Okay. WHO has been, give the recommendation to member state on the deletion or remove of ATT test from lot release, and that is based on the discussion in the ECBS in 2018. This is, as I said, the WHO recommendation is a scientific-based advisory in nature, and it needs the national control or national regulatory authority to put it into the national legislation. That means they probably need to revise their pharmacopoeia or revise their legislation according to WHO recommendation. Also, they have their authority or right to maintain it, based on the situation in their country. The basis for WHO to remove this test is implementation of GMP. in the manufacturing processes, and the completely validation for manufacturing processes and the quality control for in-process control or final product control, or even control for the source materials for the production. That's the critically important for maintaining or to ensure the quality of the product rather than only do the ATT test at the end. I think WHO is open for any member state regulators or industries, and to request their need and what kind of if they need any assistance from WHO scientifically or regulatory issues, they can communicate with us. Probably we can have more occasions and to advertise WHO's position on the removal of ATT tests through any channels. We have been published our position in scientific journals as well. And also, as I said, I see MRA meetings, they organize regularly every 2 years. And through that situation, probably we can have a complete discussion there. You can express your concerns or your questions there. We can have a discussion and reach consensus during that discussion.
Thank you very much. If I may add something to that, so I understand, and what is clear when you read the WHO guidelines, that the position is very clear about ATT. So you would complete Gautam's question, probably what can we do more altogether in order to promote WHO's position, which is probably quite a huge task for the coming 2 or 3 years. But I am quite convinced after what I heard today that we will get there, because everybody is quite concerned about it. And everybody is much concerned, as we heard from Dr. Sunil Goel, about the harmonization for also trade questions and easy trade questions over the whole globe, because the vaccines and other pharmaceutical products, they go in different directions in the world. So we are all interested that this exchange of products, and particularly the access, is increased the much possible between all countries, including the low and middle income countries, where we at least also at IEBS are very sensible for. So I see here a question to all speakers. Apologize if this has been mentioned before. You see the question. Dr. Ika Erwinda, Yola Ika Erwinda, is curious in case there is any adverse effect following immunization. What kind of tests should be carried out to ensure the safety of the vaccine? If I may add something personal there, I think here we have the always in the eternal discussion. What can you do, for example, what can you see in 100,000 subjects for a vaccine that will be used in 10 billion subjects? So who will answer that question? Probably WHO before. Dr. Lay.
Thank you. Yes, WHO have a special group of scientists. They're working on the investigation of any AEFI. That's the adverse event following immunization. And also WHO also publish a memoir, and where they provide you questions or procedures you should follow to investigate the causality of any AEFI. And you can get that information from WHO website, or I will look for that, because I'm not directly working on that, but I can find out the link and maybe provide it to the chat box here, and which they can give you some information and guide you how to consider the causality investigation. But from my understanding, the laboratory testing of the quality of vaccines is not the critical step for the causality investigation. There is a long procedure you should follow, but the laboratory testing, it's only one of that, or maybe the least important part of the investigation. Over.
Okay, we have two questions posed by Dr. Sunil and Dr. Pradeep. Sunil first. Sunil.
Hello, Yoris. It's not a question. It's regarding harmonization of the pharmacopoeia requirements. I do agree with Diana Agree and about WHO publication of deletion of abnormal toxicity test. But what we have experienced is different countries have different regulatory requirements, different legislations, and different pharmacopoeia requirements. So even WHO recommends that this test is not to be performed. But in particular case, when that country is following their own pharmacopoeia, so WHO's legislation doesn't come into force. So our thrust should be on individual countries' pharmacopoeia or regional pharmacopoeias, which should say that abnormal toxicity test is now no longer required.
Okay, thank you for your answer, Dr. Pradeep Das.
Hi, everybody. I think everybody can hear me.
Yes.
Yeah. So I echo with what Dr. Sunil has said, that this is the classical case where we got the waiver in India for related to abnormal toxicity, and in spite of WHO also, as per TRS 1016, they have very clearly mentioned, which I have mentioned in my chat, that there is no longer required for existing monograph and other upcoming monograph. But however, when we are sending it to the different countries, we feel the same, but we still perform the abnormal toxicity test. So in case we can harmonize with multiple country-specific monographs, other countries also sometimes they take the WHO as a guidance, but however, when it comes to the ATT, they recommend. So in this scenario, we request from the committee is to harmonize full force for the removal of abnormal toxicity test. In case for any one of the member mentioned that adverse reactions are happening in the field market, but to prove that if abnormal toxicity can be done over there, you cannot find it in guinea pig and mouse model in a similar situations what we are seeing in the humans. So abnormal toxicity test cannot give a directly proportional to AEFR. It is the things for the specification, and it is the national regulatory or WHO to decide what is went wrong for that particular individual. And there are cases wherein 1 in 1 lakh or 1 in 10,000 or this will, there were cases incident. There are multiple reasons for this. It is a separate committee who can look for the adverse reactions. That's all from my side.
Thank you very much. Very informative and interesting. So I see no other questions except the answer of Dr. Krapkova on the question of Dr. Yola Eka in Russia. If there are adverse effects, the samples of the batch are sent to an official laboratory for testing. Then there is another question that just arrived, a general question for all regulatory panelists. Even if the test is waived for the manufacturers, will the test still be conducted upon importation? And if so, what is the rationale? So I think the answer is somewhere already in all the answers we heard from regulators and particularly also from our Indian colleagues. Is there someone who likes to answer to that?
I would like to give my opinion about the pharmacovigilance system. So as far as the AFI in India are concerned, we have the system in India to collect and track the adverse events following immunization. There is a complete unit, immunization technical support unit in the Ministry of Health and Family Welfare, Government of India, and Pharmacovigilance Program of India through its ADR monitoring centers also collects the spontaneous reports on account of any adverse events due to the vaccines. And today we have 442 adverse drug reaction monitoring centers set up across many states in India. Thank you.
Thank you very much. If there are no remarks or questions anymore, unless I did not see something. Laura, did I see everything?
Yes, I think so, Joris. I guess that's we can maybe see if there are some additional remarks from the other colleagues from perhaps Japan, China, Indonesia, just really to understand. So we saw different perspective, different successful cases where ATT has been partially waived or partially deleted. There is a lot of openness in many of those countries to take some concrete actions. It would be nice if to hear from them if this dialogue today is actually something that has been found useful for the future implementation of the changes.
Who can give an answer to that? I can complete what Laura says, that compared to what we heard in Bangkok in 2019, December, it's almost 2 years ago now, I feel there is quite a lot of progress, at least progress in the mentalities. And then we come to the translation in the field. We all know it needs time, but I have the feeling we are on the good path forward. So if there are no questions anymore, I think we can save some time with this session. I would propose then to close it and to hand over to my friend and colleague, Von Kubiak.
Thank you, Joris. Perhaps we might take the break that we didn't really take before, if it's okay with everyone. So we have been on for some hours now. So yeah, if everybody is okay, and also if Bogna is okay to have a 15 minutes break and then we reconvene.
One word, I'd like to thank you, particularly Laura, and the whole team of EFPIA for organizing this, and we were very happy to be part of it. So you know I have to leave now, and I wish you all the best and hope that we will meet soon again, all of us. Bye-bye.
Joris, on behalf of EFPIA, I'd like to specifically say thank you to you for moderating this session, for joining us today, and also to the whole IABS colleagues for their collaboration with us in preparation for this today. And I hope it's not the end of the collaboration also, but thank you very much.
Thanks a lot. I hope and I'm convinced there's only a start. Bye-bye.
And then to everyone, I think, so as Laura has said, we take 15 minutes, so that would be 40 minutes past. So wherever you are, it will be 40 minutes past the hour, wherever you are at the moment. And we look forward to seeing you then again.
Welcome back to our workshop. We are actually entering the collaborative session where we aim to discuss about what we heard so far, understanding other participants' opinion, experience, if they want to share that with us. And then we will make sure to try to find some common solutions or some activities that we can put in place to continue the support to the deletion of the abnormal toxicity test. We will be helped in this important exercise by Von Kubiak from the International Alliance for Biological Standardization, the European branch. Von has over 40 years of experience in global animal health, with a primary focus on the development, licensure, and maintenance of global veterinary biologics. He has helped develop, improve conventional and innovative immunobiological for the veterinary medicine products for all the major species during his career, with position in R&D, quality assurance and quality control, regulatory affairs, product management, and commercial operation. Before he retired from the position in regulatory before retiring in 2019, Vogn spent the last 17 years with Zoetis, where he held management position in regulatory affairs, biological process development, and biological and analytical development. So we have an expert in all the manufacturing process, including regulatory affairs. And it's actually very important for us in order to make sure that we are actually covering all the aspects of the deletion of the abnormal toxicity test. Vogn, thank you very much. And the floor is yours, but I also encourage the participants to participate concretely to this session. Thank you.
Great, thank you, Laura. And listening to the introduction and the over 40 years always makes me feel a bit old, but hopefully there's a little bit of experience that comes with that. Before we dig into the collaborative session, however, I'd like to first see if there's anyone else in the audience that would like to add on to the conversations from the previous session and share any of your local experiences with the removal of the ATT in either your country or your region. So I think you can either send a chat or you can raise your hand. But if there's anybody that just wants to add on to what was said at the previous session, since we have a few extra minutes, please feel free to send a chat or raise your hand and we can hear from you. I'll pause for a second. Okay, so I don't see anybody raising their hand or chatting, so we'll go into the next session if that's okay. But certainly if anyone during the course of the next session wants to share some of their experiences, this is supposed to be very collaborative and it'll be great to get any insights possible. So I'll open our collaborative session now and remind everybody that the overall goal is to identify concrete actions that we can take to speed up the global elimination of the ATT and any related batch animal testing. And again, feel free to communicate either through the chat, which seems to be working quite well, or through raising your digital hand. And please remember to keep your video and microphone off until you're recognized to speak. So as I was preparing for this, I was thinking about some of the sort of questions that come out as we think about this animal toxicity test removal. And one of the first questions is specific to the actual ATT. I think we've heard a lot of speakers say that the ATT is completely irrelevant in terms of assessing the quality of a product. But by the same token, there's been a couple inferences by speakers or in the chat room that maybe there is some relevance. So to start it off, I would just like to confirm and hear people's comments. If anyone believes that there is a technical justification to use the ATT and particularly using it as a batch release assay, just to make sure that we have all of those points out first and we can move on to some of the other discussions. So I'll pause for a second and listen to see if anybody has any comments relative to potential value for the ATT.
Vaughn, it's Kirsty here. Maybe silence is a sign that people are searching identify the value. So I do hope we have people online who are willing to speak up, because we cannot identify what the bottleneck is until we hear more on where people are finding the value.
Yes, and I was being patient because many people type as slow as I do, and that being the case, it takes a little bit to get that in. But as said,
sorry, Vogna, we have Octavio.
Oh, okay, great.
From Brazil. Sorry.
Okay, Octavio.
Oh, thanks. Otávio Presgrave from Brazil, from Bracklin. About your question about the value of the ATT, I think that there's no way to perform anymore this assay. Our reality, our experience in my institute, in the institute where I work with, is the National Institute of Quality Control and Health, which is linked to the National Surveillance Agency of Brazil. We have already just published an article showing that during the last 15 or 20 years, I don't remember at all the results were negative. And also, if we look by the statistical point of view, it's completely unbelievable that two guinea pigs and five mouse can show any statistical significance, and in order to really prove about the existence or not of the toxicity. So really, my group, we have two people here of my group, Lilia and Christian also, and which are co-authors of this article. We really don't believe it. There's no way, there's no value for this anymore for continue performing, as I say, based on our experience in our institute.
Okay, great. Thank you very much for those important comments. Does anyone else want to share their thoughts on this before we move on? I hear a broad consensus that the ATT is technically no longer justified. But I see that Pradeep Das has a comment as well. His hand is up. If you would like to share that with us, that would be great.
Hello, everybody. Am I audible, right?
Yes, you're on. We can hear you.
Yeah. So I fully agree with Octavia on this. During my past 15, 16 years of career, abnormal, I'd never see a failed product which is showing abnormality toxicity as a failure. And being the test injected in intraperitoneally, the route, whatever it is mentioned, it will not give you a technically reason to produce a toxicity. If you want to, that's why this test is unbelievable. Yes, in 1980, 90, and 1900, it was a method where the phenol is majorly used. But the current in the GMP systems, if you, I have also personally conducted experiment. If a sterility failure, for example, any batch failed in sterility, you take the sample and inject in the to perform abnormal toxicity. The batch will pass for ATT. But the subject batch is failed in sterility. So in this scenario, your abnormal toxicity is passing. But can you release a batch? Yes, its mind sterility is failure. So this is absolutely not a representative model to test the abnormal toxicity. Yes, it is there fundamentally from the beginning. Now, after 2015, WHO, Edqm, everybody has came together to remove this test, and we need to still pursue that. This test will not give you any scientific rationale to prove that it is having some identify any root cause to one particular problem. I have personally seen of last 16 years of my career in quality control, you will not find a single product failing in the abnormal toxicity. In spite of you take any sterility failed sample and test it, that I am damn sure it will pass, until unless it is not a pseudomonas. That's all from my side.
Okay, thank you very much for that comment. And I think you've sort of summarized the belief of all of the participants. But I do want to make sure that we have, a consensus that the test does not offer technical value to the batch release testing. And I'll just pause for a second to see if anyone else has a comment. And if not, we can go on to discuss, ways forward to move from a lot of regional or local successes to global success. And certainly a number of the speakers have made the point that while there may be local successes in the absence of a global success, they're still having to run these tests. So I'd like to sort of open it up now for comments and thoughts relative to how do we take these examples that have been shared of country or regional successes and use those as a foundation to finally get a global removal of the ATT. Certainly there's been a discussion about harmonization, and we should talk about that. But there also should be other questions and discussion around what other things we can do. Harmonization on a global basis will take some time to move it forward. There may be some skills that are already present that can help move local programs forward to achieve a global success. So again, I'll pause and let people raise their hand or send a message. I noticed that within the chat, there is a question that says, can we hear from agencies that still require the test on the rationale for keeping the requirements? And I think that maybe we could open it up to some of the agencies to share their thought processes in these. And again, please feel free to either send a chat or to raise your hand.
Again. I guess that we still, I can try to see, we have some, I can tell you what kind of regulatory authorities are still in. So in case they want to, I see that there is Carmen. So I give the precedent to Carmen.
Okay, good. Hello, Carmen.
Thank you. Sorry, I have no regulatory answer now, for I'm a little bit concerned that this test survives and survives and survives. But the question is to OIE to see if we can have a push on harmonization. Is there an overview at WHO level, for example, which countries have now deleted the test and which have not, so to see the progress from year to year? And then maybe this is of help for the reluctant authorities to delete the test. I know from some colleagues with some talks over the coffee, there is also sometimes political pressure to maintain the trial. So if a batch fails, then the first question is, did you test the batch in animals? Yes or no? Maybe this is an aspect which is also there like an elephant in the room.
So very good, Carmen. So I haven't seen an OIE representative, but there's quite possibly one there.
Sorry, a WHO.
Oh, okay. Yep. No, but I think it's actually a valuable discussion, even more broadly, because there certainly are some learnings on the veterinary side as well. But maybe our colleague from the WHO would be willing to comment a little bit on the availability of some sort of a status report that really puts all of it in context as far as progress. Please.
This is Denali, and I'm working in WHO, but we do not have the statistics of which country they are still keeping the request of doing ATT test. But in general, we organized the Edqm meeting. That's the International Conference of Regulators, and where we pass the message of WHO recommendations to the regulators. And as we heard from Carmen, and some countries they may have a variety of reasons to keep the test. Either it's technically or maybe politically. I think that's the reason some countries they keep it. I think the most important for us, we may propose, even to WHO or any other, global organizations to organize kind of workshop and to really to listen to the concerns of the regulators. What is your concern? Probably we can explain to them what is the scientific basis. We made the decision to remove the test from the WHO recommendation, or maybe some regulators like US FDA or Edqm, they can share their experience. How did they do to remove it and remove the test from their pharmacopoeia? I think their experience also may be very valuable to the regulators. Now they are still keeping this test, which provide no technical or technical information to the decision or to the quality of the product. I think probably we can think in that way, and to really to listen to the regulators and discuss with them and pass the message. What is the scientific basis for the decision we made?
And so with this, thank you very much for those important comments. Would this be something that would be initiated as far as having this stakeholder meeting from the WHO? How would that next level of conversation occur?
I think maybe if we can share the meeting report and with WHO, or we can send formal request to WHO and then ask WHO to organize kind of workshop and with regulators where they have been already removed the test, and also especially the one who is still requesting this test. And we can have a dialogue and a workshop with them. Either it's an online workshop, maybe face-to-face is more, more valuable, to have rich consensus. Virtual meetings sometimes, people they may not have a chance to open their mouths and to express their concern. Actually, maybe face-to-face meeting is more valuable, from my understanding. Thanks.
Very good, thank you. There was also a suggestion in the chat group of bringing this topic to the ICMRA and having it on the ICDRA agenda in 2022. And again, I think that the more different environments where this is discussed, the better the traction will be. While we're waiting for the next comment, one thing that I picked up from the presentations is that it sounds like there's quite a bit of information that's been assembled over time where local country-based colleagues have worked with the regulatory authorities to really build a case and help justify actions to remove the ATT. And I wonder if these resources that are used are broadly shared, and maybe they are, but I heard a reference to assembling and submitting white papers. I heard discussions around the type of data that have been used to offset. And I'm wondering if there isn't also a value in some sort of a common repository for these types of documents to help facilitate continued discussions, especially with reluctant authorities or politicians for that matter. Is that something that's already available? Is that something that people would see a value in having access to? Curious.
Well, I just jump here.
Yep, please.
The discussion that we had starting from 2015 through the PAA workshop that Catherine was mentioning early at the beginning of this workshop, and further the discussion that we had along all these years brought to the sharing of all the key publications that have on the topic to directly to the key regulators or industry stakeholders in some of the key countries. It's not possible, that's what I understood, to share the specific data of the batch release within the various countries. That's an argument that's a question that we made to some regulatory authorities just to really help, not maybe at the global level, but at the local level, to show what has been submitted, how it has been submitted, or to create internal local guidance in order to help the local stakeholders, basically the local industry and their national control laboratories to understand what kind of data needs to be submitted and what is approved. And as Anthony also wrote in the chat right now, the Indian case can really be used as an interesting case study. As Sunil and Pradeep mentioned before, the dialogue and the exchange that they had with the Indian Pharmacopoeia Committee was definitely useful to allow both the regulatory authority to be confident in the change, but also industry itself to demonstrate the quality of its production process and its product. So the idea is, if we can really learn from the other participants here, if a similar approach could be beneficial, where the majority of the doubts are still in place. We have seen also what China has done, that after a lot of years of dialogue, they managed to have a little bit of language into the Chinese pharmacopoeia. So we know that dialogue is the base. It's important to understand if we need to go again using this approach and continue the promotion of the dialogue through WHO, through local specific events and sharing, or if we can hope for a big giant step and say, okay, we agree the test is completely useless, let's remove it. I know that it's not possible. We again, the Chinese colleagues from RDPAC, they mentioned that there are some situations where local manufacturers or local realities are probably yet to achieve a level of quality system implementation or even pharmacovigilance at the level of the country that is probably slowing down this type of discussion among the local stakeholders.
Okay, good. Thank you very much for that. And certainly the concept of a case study may be something that helps stimulate the conversations. There's another comment in the chat which I find interesting because it takes this to a different tact. It takes it away from the technical and it takes it to the reality of what's going on at the authorities. And it says, what would you advise if the local authority is simply not prioritizing ATT deletion, even though they generally follow WHO recommendations? They don't think that the ATT deletion is something that should be considered urgent. So the inertia is not around the technical aspects. The inertia is around prioritization. And I wonder if anybody has experience with that, or anybody can share how to raise the priority of addressing this issue within countries where maybe it isn't such a priority. Again, I'll look for anyone in the chat or a raised hand. I mean, I can see this as being quite a potential issue. people have talked about how long they've been working on this, and people have talked about the challenges of the timeline and possibly the frustration of the timeline. It's one thing to have a strong technical argument. It's another to get the visibility necessary to make progress. I see a lot of hands up all of a sudden. This is sort of exciting. Let's go to Sunil first, please.
Yeah, thank you very much for giving me an opportunity. I just would like to add my experience with reference to Indian context. It's a number of years. I actually have a past experience of Indian National Control Laboratory before switching over to private industry. So we had been performing abnormal toxicity test there for a number of years, but no outcome, no failure of abnormal toxicity test was observed. However, we had to comply with the pharmacopoeia. Time and again, we had to perform the test. Even when we received samples for investigation of AFI cases, then sterility, abnormal toxicity, biochemicals, and other parameters, every time they were performed and couldn't come over to the conclusion what could be the reason for adverse event. So these things, number of samples testing every year paved the way that it's not going to give us any good output. So that was one thing apart with the regulator. But on the other hand, manufacturers, like we manufacture over 5,000 batches per annum, perform abnormal toxicity, not a single failure, just wastage of animals. So it was in 2014 and 15, we used to have a collaborative workshop of National Regulatory Authority, National Control Laboratory, Indian Pharmacopoeia, and the manufacturers. This point was put forth. Initially, there was a lot of resistance, like things had been going from year to year. So there was a lot of resistance, but then it was a big challenge how to break the ice. Then we gathered the amount of data all the manufacturers had and the data generated by National Control Laboratory, not a single failure in abnormal toxicity for a number of years. That paved the way, initial thought. Second point was an open environment in India between the regulators, National Control Laboratories, Indian Pharmacopoeia, and manufacturers was second soft ground, which could help this seed to grow. So that paved the second way. Third was all the manufacturers. Now India has a good number of vaccine manufacturers, say about 20 plus. So that majority also helped. That we all got together, we had a strong voice, which had a discussion with the regulatory authorities. That gave another point of thought. Next was impetus by other non-government organizations like People for Animals, HSI, etc. They gave impetus. They were on our back. Yes, you move ahead. We are supporting you. And next point was not to reinvent the wheel. Once European Pharmacopoeia published its recommendations, WHO gave its recommendations, then gathering all these data together, we went to the door of NRA and in the Pharmacopoeia Commission, though it took time for one meeting to another meeting. And not only this, we had certain issues within the manufacturing group also, because there were different ideologies. There are different types of GMP environment in each of the organization. One was having a very high level of a GMP, he was comfortable, but other person having a low level of GMP was not comfortable. But then keeping in view the regulatory audits were taking place quite frequently within the country, when in order to comply with those audits, manufacturer had to upgrade its GMP system. Once all the GMP levels were at par, the National Regulatory Authority was convinced, yes, now we can allow them for, initially it was a waiver when we had to perform abnormal toxicity only up to consistency level. And if it is to be performed by National Control Laboratory, it should pass. But that waiver was again like the very first opening discussion today, post-approval change. It was a very lengthy procedure. So though we had substantial amount of data, but the time taken is quite long. And for an exporter like Serum Institute, we had to apply to 170 countries to apply for a change in abnormal toxicity for a waiver. It was a big task.
Yep.
So all these things then but we patiently moved on ahead, and what we got was that united we stand, divided we fall. We remained united. We got together with the National Regulatory Authority, with Pharmacopoeia Commission, and all these things. Finally, they gave way to us. And in July 2020, we could achieve final deletion of abnormal toxicity test from the human vaccine monographs. So that's what I wanted to share with you.
Okay.
That nothing is impossible. Things are possible, but we have to frame a proper path to it, how to give direction to our objective.
Good.
So it can be put into a very good case study as well.
Yeah.
Perfect. Good. Thank you very much. I'd like to move on to Dr. Youngbach, who also has her hand up.
Yeah, thank you, Ron. I come back to my favorite overview, and what Sunil said was excellent. For this was excellent. The work, it takes a long breath to persuade all stakeholders to delete the test, and there are some conditions needed. But what Laura said already, we cannot publish the test results. Yes, of course. But what is possible is to summarize the number of tests and a list of vaccines tested without mentioning the trade names and whatever. Then say we have tested 20 different vaccines against this and that antigens, all inactivated. We have tested in general some thousand batches and nothing happens. And this is something which everybody can make. And then we do not have a damage of confidentiality of the data. But these kind of overviews should be really of help. And if then it's really visible that hundreds and thousands of batches were tested and the outcome was always that nothing was detected, then I think this can persuade a lot of people more. And this big issue which has been done in India is something which not in all countries can be performed due to the lack of human and resources sometimes. And therefore it may be of help if a number of countries all over the world show how they had approached and how they managed it.
Great, thank you very much. I noticed that Lisa Plitnick has raised her hand as well. Lisa, would you like to share?
Yes, thank you. So my question is around, and I've asked this, and I think maybe I'm not asking the question clearly. My question, and I'm not clear on, is for the countries that have removed the requirement or allow a waiver given GMP data, when the products are sent to those countries, a lot of times they will test it again upon importation. And so I think, I know that China does that, and I just am not clear. So for the Indian pharmacopoeia, the way it was written, it also sounds as though that testing is still conducted. So even though the manufacturer's requirement is waived, are the tests still being conducted in the countries? Because in my opinion, while it's great that the manufacturers no longer have to do the test, it can be very complicated if the individual countries run the test and get a failing result, and it can get very complicated from there. So that's my question: is are those requirements still in place in the Indian example? And if so, what is the rationale for keeping that requirement in place for testing when the vaccines get to those countries?
Yeah, if I clearly understand your question, first of all, whatever vaccine manufacturers produce vaccine for Indian domestic sales, which are released by the National Control Laboratory, does not require abnormal toxicity test to be performed. Number two, whatever supplies which are being made to UNICEF from India, that too also doesn't require abnormal toxicity test to be performed. But there are a few countries who directly import vaccine from India. For them, we need to perform abnormal toxicity test based on their pharmacopoeial requirement.
So,
am I clear?
Yes, that was very clear, but I think the question really took it one step further, which is even though there may not be a requirement for the manufacturer to do the ATT test, there may be countries that do the ATT test, even though it's not a requirement for the batch release from the manufacturer. Maybe it's a requirement by the authority before they will release it. Is that the correct interpretation of your question, Lisa?
That's exactly what I'm asking. Thank you.
Yeah. So maybe somebody that has awareness of the decisions at the country testing level by the authorities could answer this question. I don't see a hand coming up. I do have a little message on time here, though.
Oh, and I was just going to clarify that in my experience, Russia has been one example, China another, and Japan. So I know we don't have any regulatory colleagues from China on the line, but maybe someone from Russia or Japan could comment.
Recognizing that it would be need to be a fairly quick comment, if there's anybody from China or Japan that would be willing to address that question, I would appreciate it.
Hi, this is Jack from China, but I didn't get that question very clearly. Can you repeat the question again?
So the question is specific to testing of a batch of product coming into China. And specifically, do the Chinese laboratories test using the ATT test, even though it may not be required of the manufacturer? Does that make sense?
The signal is not very good. I missed the first part still.
Okay, I'll try one more time. So in the situation where a vaccine batch is shipped to China, the authorities will do some testing on that vaccine batch. Will they do ATT testing on that vaccine batch, even if it's not required for the manufacturer?
Yes, yes, that's correct. That is the current practice or requirement by the China CDE and NFDC based on the China Pharmacopoeia. NFDC, which is the formal test laboratory by the health authority, will do this QC test before each batch release.
Okay, so how do the Chinese handle data where they're testing it for ATT? Do they ever find unsatisfactory results? And then how is that addressed with the manufacturer?
That's a good question. For biologics, for most antibodies, because they only require mice and this test is pretty straightforward, most of them can pass the test without any problem. In some special case, there's some findings from the test. Please be aware that any findings, not only the adverse findings, if there's any findings different from the saline control group, they will judge it as positive. So for some products, we did see some problem. So we will, as a manufacturer or the sponsor, we will be asked to provide any evidence to show that this is because of the product components, but not because of the contamination. If we can provide this solid evidence, NFDC may retest it and pass your product.
Okay, very good.
And for vaccine, especially for COVID-19 vaccine, because these vaccines based on different vectors, a lot of the sponsor has no confidence that we can pass the test directly. After communication with the sponsor to do the exploratory test, make some modifications for the test condition.
Okay. Very good. Thank you for that very helpful answer. I now realize that I'm past time, so I would like to thank everybody for your thoughts, your comments, and the discussions. I believe they were very helpful. And I now would like to turn the meeting over to Rajinder and Philippe for final remarks.
Thank you, Professor Kubiak, for conducting a scintillating session. This was, I think, one of the finest Q&A session I have attended for a long time. In fact, it is, let me just start the video, and yes, here we are. So it's my honor to say a few words as concluding remarks, addressing such a learned audience representing the whole world. And I must start with a statement that in last 20 months, probably this is the first meeting that has been talking about animals and not human beings and not COVID-19. So it was a pleasant change to be participating in this and listening to very engaging and enriching interactions throughout the sessions. So my congratulations to all the honorable speakers for crystallizing their thought processes so vividly that ATT being a non-specific, lacking relevance and not reproducible test is already rendered obsolete, and thus bringing the urgent need to the fore to make all-out efforts to ensure that there is a global deletion of ATT once and for all. The efforts of US, Europe, WHO, Brazil, and latest, I would say, India, and many other praiseworthy countries are praiseworthy who have already deleted ATT starting with 2015, then 2018, and then some of them in 2019 and 20. Here, I would like to bring the reference back to the points which were raised by both the keynote speakers, my friend Thierry and Catherine, on the complexities which are involved in the vaccine manufacturing processes and the testing part of it, and particularly when Thierry was referring to the polio antigen. With one antigen, 500 changes are required in the dossiers globally. So I think this assumes a very high significance that since ATT lacks scientific relevance and its deletion is not basically compromising the safety of the product. So I think the fourth R, which was proposed by both the learned speakers, whether you call it rely or remove, I think that is what is going to be, should be implemented. In fact, even Kirsty in her opening remarks had said that a batch which was already released for EU would have to be tested for abnormal toxicity in other countries. Thus, I think it becomes very apparent that it is justified to completely eliminate the abnormal toxicity test from pharmacopoeia. These views have very well been endorsed by our learned regulators. I heard Dean Smith as also Dianling, and they very categorically said that the reliance on ATT is not anymore required because the general safety test has failed in its very objective, and there is no scientific rationale. In fact, Dianling has been very, very precise that WHO has been working on this since 1999, and finally the deletion was recommended in 2018. And what is important is that once the production consistency is demonstrated, you don't really need to rely on this kind of irreliable tests. And I think I would also go with this statement that the validation of manufacturing processes is what is required to ensure that there is no compromise on safety of the product. I totally agree with his thought process on that. And Robin had very clearly stated that there is no product requiring ATT even for COVID-19, whether at the time of release or at adverse event, because it has no impact. Finally, I would also like to bring here the point which Joris, while moderating at the close, he said that there is a change in the mindset which is happening globally, and I think it is a very, very good beginning to an end of ATT once for all. So my point here is that I would like to make, it makes absolute sense to permanently delete ATT globally, which would not only save millions of animals, but would also reduce time to market and ensure faster availability of the products. And in addition, of course, save some millions of dollars to the manufacturers, which ultimately can be converted into the price affordability to billions of people who are looking for the vaccines. And we have been talking about the vaccine equity, and especially that in LMICs. So with this, I would like to frankly congratulate and thank AFSA, HSI, and EFPIA, and especially the three young ladies, Laura, Kirsty, and Tatiana, for organizing such a great event with a great zeal and passion. And I would like to wish good luck, and would like to pass on the mantle to my friend Philip. But before that, I think there are 3-4 action steps which just came to my mind, and I thought I must share with you that going forward, what all can be done, because it was suggested by Dean to raise it at ICMRA level. And so ICMRA, ICDRA, and other regulatory agencies together. Philip had said that we need to rationalize with the support from DCVMN. So here what I would like to propose is that, as was recommended by Dian Liang also, that a workshop probably organized by WHO, but we can collaborate with BMGF, with like-minded parties like AFSA and HSI and EFPIA, all of them together. But DCVMN can definitely take the lead in this, because the focus is on manufacturers and countries in developing region. So I would say that we would probably put together a proposal, share it with WHO, and with their good offices, we can bring regulators and manufacturers together on one platform to make sure that there is harmonization, which is being very well conceived and perceived by everybody who has spoken on this platform. So with these few words, I would like to pass on the mantle to Philip. Thank you.
Thank you, Dr. Suri. Yes, I would like to thank the attendees, the high level of engagement, also the organizers. So thank you very much for that event. So just like to, I know we're past time, so I'll try to be brief. Would like just to remind you the foundation mission in terms of trying to enable the development country vaccine manufacturer to produce vaccine sustainably, and at the end, so we can increase the vaccine access. And that's our most important priority. I think during that meeting, it was extremely important, all the discussion around the standardization of the methods, and it's totally online in what we would like to implement in terms of having more efficient ways to operate in the DCDM, cost-effective, relevant. There's a lot of challenges to implement an industrial environment in the low-medium income country. And obviously, we don't want to torpedo them with non-relevant activities. It's extremely important for us to make a space so we can really use the newest technology and move past anything that's outdated or inefficient in terms of practice. I think we had a very good discussion about the standardization of the methods in general. So I would like to, I'm sorry if I'm a broken record, but I think we also need a standardization of the removal of the methods. I think there's a lot of things we can learn from each other. That brings a concept of that networking of manufacturers that Dr. Suri is really championing with the DCVMN. And also, I think we understand better now there's also a need for a networking between the regulatory authorities. So they can also exchange that information overall, so we can share our experience in terms of and success in terms of removal of those tests. And it was very encouraging, the level of discussion we had between us in that meeting, between different countries and different realities. So I also would like to mention Thierry Gastineau, a former colleague of mine at Scientific Pasteur, for the last R. I think we all have our proper definition of that R. In my case, is reaching out. So in terms of networking. And so I think we all coming from different sector of the industry. So I think we all understand our duty is really to continue continuous improvement and while providing access to safe product. So thank you very much.
Thank you so much, Philippe, and thank you so much, Rajinder. You both have summarized today's discussions so well. And first, I'd like to look at yourself, Rajinder, and thank you for your proposal to put forward a sort of kickoff discussion or to move forward to take something to the level of the WHO, working across the different stakeholders. So we would definitely take that forward. We would welcome that, and we will work with you as you feel fit. So I'd like to use this as an opportunity from Laura and myself to sort of close off the meeting and also discuss some next steps. Now, what stood out for myself today is it's taken a long time to get to where we are today, but recently we've seen many positive steps, and for the first time we're sort of starting to see a lot of changes taking place. And I've been involved in the sector for a while, focusing on the animal research and alternatives and the uptake. And I don't think I've ever been part of a discussion before where I've seen so many of the different stakeholders all wanting the same thing. And I really welcome that. And I feel that that is what's going to bring the change. The political pressure, the societal pressure, the science behind it will be what brings it forward. Now to discuss next steps, just as a general from ourselves, from Laura and myself. So we will be preparing a report as follow-up to this meeting, but the report will of course take a bit of time. So in the time taken to develop that report, we would welcome any feedback that you have, any scientific papers that you've referred to or that you would like to share with us, that we can take it into account, or any comments or statements that you did not make in the chat today, or that you haven't brought up during the discussions, or you feel weren't brought up in the discussions. We'd like to take that into account as we move forward. Already next week, we'd like to share with you this video and any of these any published papers that you may be sharing. We will of course share other documentation like the bio of the panelists. I'd like to still say a thank you to everyone. I don't want to take any of Laura's comments that she may wish to share in the next steps. So I'll hand over to herself, but I'd really like to then have a moment to still be able to acknowledge some of the work that took place today.
Thank you, Kirsty. Very briefly, you said already everything. We are, as AFSA, Animal Free Safety Assessment Collaboration and Humane Society International, we continue to promote the local dialogue beside this workshop. We had a fantastic start today. There are a lot of information and a lot of reality that we would like to hear more about. And we already realize about some of the challenges that there are still in place, and we will all together be happy to overcome all of those. As Kirsty say, stay tuned for our final report, and we hope really to continue the collaboration in organizing this meeting together with WHO and all the relevant parties. Thank you very much also from my side to all of the behind the scene people that help us, and also all the speakers and all the participants today. Kirsty, for you for the very final one.
Yes, and to build on what Laura was saying, first of all, we couldn't have moved forward without the level of speakers and the panelists we had today, but also the moderators who did an excellent job. And I'd like to personally thank all of them. However, there's also the work that takes place in leading up to any meeting, and we'd like to just acknowledge those people who were part of it. So we had a small steering team who helped and worked with myself and Laura, but at the same time, we had help coming from our various colleagues. So we'd like to really just put their names down on paper here to acknowledge and say thank you personally to them for the work they've done. And then just as a final word from myself, I'm a South African, and I often finish off a discussion with a quote of someone I admired quite a lot. So I know this happens, many people may use similar quotes from the same person, but I do have a lot of respect for Nelson Mandela. And one of his quotes, which I feel fits very well in today's discussion, is that it always seems impossible until it is done. And I think we're very much moving on the way to ensuring that it is done. So Laura, any final word from yourself? Otherwise, we will
adjourn the meeting.
Sort of wave goodbye and thank everyone for being there today.
Thank you very